Hello everyone, welcome to this session 3 of the Thesis presentations. So just to refresh your mind, the students, we are going to watch 3 presentations from 3 students. They have 15 minutes to present their thesis, and then there will be 15 minutes of questions. Of course, our jury has the priority, which we will present shortly, and then we will ask if there's questions from the public, and if we still have time, if there's questions from the YouTube chat, because these presentations are live streamed on YouTube. So let me introduce your jury for this session. We have Professor Fabio Rindi from the Polytechnic University of Marche. Then we have Dr. Jasper Dierik from the University of Ghent and the University of Brussels. And then we have Dr. Francesco Manuel, also from the Polytechnic University of Marche. Without further ado, I think you can go. You have 15 minutes.
Thank you. Good afternoon, everyone. I'm Chiara Marulli, and my thesis is about optimizing nursery conditions to promote seagrass production for large-scale restoration. This was done in collaboration with Central Queensland University, Australia, and my supervisors are Emma Jackson and Andrew Ostrowski. First of all, what are seagrasses? Why are they important? Seagrasses are marine plants, and they are among the most productive ecosystems on the earth. They provide habitat and food for marine life. They also improve water quality, stabilize sediments, and protect coastlines. And they also contribute to nutrient cycling and carbon sequestration. However, they're facing many threats, such as anthropogenic activities, pollution or coastal development, but also climate change stressors, such as marine heat waves. And therefore, they're globally declining. One possible solution could be to use a seagrass nursery to provide seeds and plants material for restoration, such as that picture right there. However, nurseries, nutrients can be limiting factors due to the reused sediment, but also the recirculation system of the tanks, and also due to the algal competition. However, there's been many benefits from the addition of fertilizers into the soil, and therefore the objective of this study was to assess the growth and physiology of species of seagrass, Zostera muelleri, under different fertilizer types, but also under different concentration of fertilizers. Overall, the objective is to identify nursery best practice to enhance the production of Zostera muelleri in order for the future to have more plants and seeds material for restoration. Why Zostera muelleri? Because it's a dominant species in the area, it has a high seed production, it's easy to harvest and easy to cultivate in nurseries, as you can see from this picture here. In my experiment, I used 2 fertilizers, osmocote and struvite. They have a different nitrogen to phosphorus ratio, and they also have a different physical form. We can see that the osmocote is in granules, while struvite is in a powder form. The pros of osmocote is that it's been widely used in restoration, and it's commercially available and it's affordable. However, this solution can be temperature dependent, and this could lead to potential over-fertilization and therefore be harmful for the environment. Struvite, on the other hand, is a new alternative fertilizer. It's a byproduct from wastewater treatments, so that supports nutrient recycling, and also has a low solubility in water, which means that can be a slower release fertilizer. The concept of struvite is that it's not commercially available and it's not affordable. Zeolite, on the other hand, is not a fertilizer, it's a soil amendment, so it acts as a carrier for nutrients. It helps to retain nutrients in the soil and also reduce nutrient leaching. So it has to be used in combination with the fertilizer in order to enhance the fertilizer efficiency. So this is the experimental setup of my study. I have 8 control treatments. I have a control treatment without fertilizer addition, and I have osmocote at low and high concentration. I have struvite at low and high concentration, and I also have treatments with low concentration of osmocote and low concentration of struvite with zeolite addition. and zeolite as treatment as a procedural control. Each treatment has 10 pots, 5 of which are unplanted to check if there was any external factors that could influence the release of nutrients. And the other 5 were planted with 2 plants of seagrass per pot. And therefore, the structure is a nested structure, which was considered in the statistical analysis afterwards. The pots were randomly placed in 2 flow-through mesocons, and the duration of the whole study was 8 weeks. So first of all, I like to collect my fragments in a seagrass meadow, which is called Pelican Beds. It's off the coast of Gladstone on the east coast of Australia. I prepared by sieving sediment. I prepared a sediment mixture, and I also prepared the pots by adding the substrate and the fertilizer individually to each pot. Before transplantation, I standardized my fragments by removing any brown leaves and also by cutting the rhizome after the 4th shoots, in order for each plant to start with 4 shoots. What I measured, morphometric changes, I measured the number of shoots per plant, and I also measured the leaf surface area, which was done by multiplying leaf length, leaf width, and number of leaves per shoot. This was done on a weekly basis for 8 weeks, and before and after the experiment, I also measured the rhizome length and the biomass as wet weight. I also checked the photosynthetic efficiency of plants on a weekly basis with a direct contour on light adapted leaves, and I also measured the sediment per water nutrient content in order to check the initial and final exposure of nutrients that seagrasses were exposed to. And I collected and filtered the water, and I sent it to an external lab to check for nutrients such as total nitrogen and total phosphorus. About the results, here is the number of shoots at shoots density. Each plant started with 4 shoots initially, and the osmocote treatments, however, at the end of the experiment increased approximately by 6 shoots per plant, and which was significantly different from the control treatment. The struvite treatments, on the other hand, were similar to the controls that are in red, and they're kind of seen a slow decline towards the end of the experiment. Regarding the leaf surface area, the graph is showing the percent change in leaf surface area, so they all over time. They all start at 0% as their initial value, and we can see after week 1 there is decline among all treatments, and that is possibly due to transplantation shock. So there was a loss of around 20 to 40% of leaves. But at the end of the experiment, we see osmocote with zeolite in blue, which increased by 35% of their initial size, and the osmocote at high concentration increased by 10% its initial size, and osmocote low increased at a less linear increase with the purple line. But all 3 osmocote treatments were significantly different from the control treatments. Struvite treatments, on the other hand, were similar to the controls. We can see a slow decline towards the end of the experiment, which is probably due to the senescent period. Regarding the rhizome elongation, the graph is showing the percent change in rhizome length across treatments. All treatments increased in rhizome length. However, we can see that they also had low concentration, high concentration, and with zeolite more than double their initial size, and which was significantly different from the control treatments. The struvite treatments we can see here were similar to the controls, with an increase of around 60 to 75% in rhizome length. Regarding the biomass increase, the graph is showing the percent change in biomass across different treatments. The controls did not increase in biomass, And we can see that the osmocote low concentration, high concentration, and with zeolite increase their biomass by 40 to 55% compared to their initial sizes. And this was significantly different from the control treatments with really low P values. And on the other hand, the struvite treatments kind of reached 10% more in biomass. However, this was not significantly different from the control treatments. This is just a picture of the same plant before and after the treatment with osmocote at low concentration. So we can see that osmocote clearly increased biomass, leaf surface area, shoot density, and rhizome elongation. Regarding the photosynthetic efficiency, I measured the yield with the diving lamp, and I standardized it by weekly mean and standard deviation, and the graph is showing that across different weeks and treatments, there was no significant difference between treatments, and this is probably due, this could mean that the nutrients don't affect the physiology of the plants, so all plants were in good and healthy conditions similar to the controls. Additionally, we can also say that the morphological changes that we saw before are likely reflecting the treatments and not pre-existing health conditions. Regarding the pore water nutrients analysis, the graph is showing the concentration of total nitrogen on the left and total phosphorus on the right. The red bar is the initial exposure, and the blue one is the final exposure without seagrass. And the green one is the final exposure with the presence of seagrass. There was no significant difference within and between treatments. However, this is probably due to the low replication and high variability, but we can still see some trends. For example, the zeolite treatments, so osmocote with zeolite, struvite with zeolite, and just zeolite, had a higher nitrogen content at the end of the experiment without seagrass present. But when seagrass was present, the content of nitrogen was lower. This could mean that there was an enhanced uptake of nitrogen when zeolite was added, or the nitrogen was more in a plant-accessible form for seagrass to obtain. Regarding the total phosphorus, we can see that the struvite treatments had a high initial phosphorus, which kind of reflects the fertilizer ratio of 10 to 23 N/P ratio. But this was also at the beginning of the experiment, so that means that struvite could be a faster release fertilizer, and this is probably due to the fact that it was a powder formulation and not in a granule formula. To conclude, we saw the benefits of adding osmocote on all the morphometric changes. But we can also say that the low dose of osmocote was effective, so there is no need for higher concentrations to stimulate growth, which can be also harmful for the environment. Also, osmocote is a cost-effective fertilizer from a nursery perspective because it's affordable and available. On the other hand, struvite did not show any significant growth compared to the control treatments. And it was also faster release, probably due to the fact that it was in powder formulation and not in granule formulation. And it's also less practical from a nursery perspective because it's not available and not affordable. Zeolite seems to have some benefits. It seems to enhance the nitrogen retention in the soil, and it also seems to enhance the osmocote efficiency with the treatment osmocote zeolite. It's also worth mentioning that all plants were going into the senescent period of the year, so we saw a decline across all treatments except for the osmocote ones, which still was able to stimulate growth even though there was a senescent period. For future consideration, seagrass nurseries can be valuable tools because they can reduce the pressure of wild seagrass populations, and they can also supply seeds and plants material for restoration. I also still believe the potential is struvite, but for further research I would use granules instead of powder to have a slower release over time. And there are still many unanswered questions, such as what are the effects of long-term nutrient enrichment on both the growth and the production of flowers and seeds. Also, when is the most effective time to actually add fertilization, or what are the effects on different life stages such as seedlings or other species of seagrass. And thank you for your attention, and I also want to thank all CMERC team and CQ University for hosting me.
Good. Thank you, Chiara, for the nice presentation. And now we'd like to give the word to the jury, and now we can start.
Thanks, Chiara. Very nice job. Very interesting presentation. So can you go back to the slide with the okay? Can you say again what is the composition of the osmoc of the fertilizer
Ratio? 4.3 to 9 NPK ratio, and also it's also good for aquatic plants, so it's supposed to be used for aquatic plants and not terrestrial.
Okay, the results look quite impressive to me because I mean, considering that you did all this at the time of your practice is supposed to be based on the senescence, it really looks like it reversed so look at what do you think is the key aspect, the key benefit that the osmocote that they have compared to the other difference in the nutrients or what else?
So, as we saw, osmocote, even though from some papers it didn't seem to have many benefits, in my experiment was really beneficial for the plants, even though they were going into the senescent period. It would be interesting to test with this struvite as granules, because that's supposed to be slower release. But from a nursery perspective, I own the nursery. Osmocote is commercially available. You can find it in garden centers, and it's really affordable. So it could be a cost-effective fertilizer, and which struvite is not, because it was really hard to find it and really, really expensive. So.
Okay. Look at. I think can you go to the slide where you showed the patterns of the biomass?
With biomass increase?
Or maybe the previous one.
Rising elevation?
No,
Leaf surface?
sorry, leaf surface. No, here I was wondering, okay, the initial decline that you observed, that there was due to what? There was a loss of some leaves?
Yeah. When I was measuring actually the first week, I was seeing all leaves coming off, and I was a bit concerned because I thought they were all going to die. But they didn't, they stayed healthy. It was really just, we think transplantation shock is they're really sensitive to changing in factors and environments. So.
Okay, I was really impressed by the increasing length of the rhizomes. I mean,
yeah.
Great.
The picture.
Yeah, I mean, that looks like a huge difference, and that is great because obviously the fragments, like the one after 8 weeks, then it means that it's something that they can break in 3 or 4 different fragments.
Yeah.
Okay, I assume all the work that you did was in the nurseries. I mean, you didn't do the transplants out in the field.
No. The purpose of the nursery is more to produce more flowers and more seeds, and then transplant the seeds since it seems more feasible to actually restore seeds than fragments. But it could also work.
I mean, something like that. Okay, then Zostramulari being that area is the species for which flowering production of seeds is predictable. Then this will be what kind of
and it's one of the dominant species in the area.
Okay, now there will be an aspect that will be interesting. That obviously, as long as the fragments are in the nursery, they're growing very well. Now, I wonder if once they want to transplant them in the field, okay, then when you don't have the supplement of the fertilizers,
Yeah.
if they grow equally very fast, or not.
Yeah, that would be interesting.
Okay, thank you.
Thank you.
Again, good job. And look, I hope that for all this, the course of restoration of citrus, and algal forest, that you look at with me, that was useful.
Yeah, I started already with the base, so that was already advantage, I would say. Thank you. Thank you.
Okay, thank you for the presentation. Your work, a lot of valuable work, I think, interesting things. Some questions. So you use these nurseries, they use fragments. Yes.
They
They also
use-
use seeds. So you work on fragments mainly.
Yeah,
But in these nurseries, they also collect seeds and then grow them in these nurseries. It's only fragments they use.
yeah. They collect flower shoots, they
Yeah.
also collect seeds, but they also collect sediment cores
Yeah.
and already put here, sorry, here, this is a sediment core that
Yeah.
was collected from the meadow and they just directly put it in a pot and they let it grow in the nursery under
Yeah.
controlled conditions.
And why did you use these fragments for your experiment and not seeds?
Because with seeds, you have to wait more time for them to germinate, and also seeds have, initially they don't benefit of fertilizer addition because they already have everything within the seeds. So at the beginning, it would not be beneficial. It would be interesting to see on the seedlings what are the effects, since they are just tiny plants, but they don't need the seeds nutrients anymore, but they need nutrients from the soil. This is.
I agree. So you collected fragments in the field.
Yes.
I'm a population geneticist on seagrasses, so for me it's always interesting to know, what do we collect? So seagrasses can be very clone as well. Did you make sure that you had like a high variation in fragments, so you covered a large area? How did this work? How did you sample?
Yeah, I sampled here in the seagrass meadow randomly. Of course, they are all from the same population, but then they were completely randomized when transplanted into the pots and the fertilizers.
Is there information on their clonality in that area?
There has been actually a PhD student who's working on genetic and different adapted methods, and there are, from what I remember, there are some differences between populations. In this case, it's all the same population, but in other areas where it's more sheltered or where it's more wave exposed or more light or less turbidity, she was seeing some genetic differences or patterns.
It's not that it was one clone that was present in that lane, you had different individuals.
Sorry?
It's not that it was one major clone, one dominant clone in that area, you had different individuals of seagrasses.
Yeah, I picked them from like.
I guess that would require a study of the population.
Yeah, it would be
No,
interesting to
okay, the
test.
largest
Yes.
plant in the world organs and the plant of the Posidonia australis in Western Australia.
And like this area that you sampled, was it like a nutrient-rich area or a nutrient-poor area? Did you measure the water parameters or the sediment?
I personally didn't,
Yeah.
because the purpose of the study is to increase the seeds and plants that you have. So using the conditions and mimic what happens already in the nursery. So they reuse the same sediment that has been used for many years in the nursery. So I didn't collect the sediments there, but when I tested for empty pots with just the controls to see the nutrients that was already present in the sediment used in the nursery. that was the goal of to check the baseline of nutrients.
Okay, and then maybe one question.
Fast.
Fast.
If it's quick.
Um, Do you think we can apply this in the field as well now? These fertilizers to enhance growth, or what are the risks? Or are there risks? If you can say briefly this.
I personally wouldn't apply it directly in the field, because in the field we have many external factors that could influence the dispersal of fertilizers, and also I applied it in the nutrients, so I wouldn't know how you would mix from a practical point of view the fertilizers in the sediment. There has been many studies in the field where they put fertilizers on top of the metals, but of course most gets released in the water column, and the amount that actually reaches the plant could be different. So I would personally just use it in the nursery in a more controlled environment.
Thank you.
Thanks.
Yeah. Okay, thanks for the presentation. Yeah, actually, yeah, I'm not an expert of the field, so my colleagues already make a lot of questions. I don't have real questions. I think you presented you were structured your presentation, and
Thank
you
you.
presented your results clearly. So, yeah.
Thank you.
Okay. Are there any questions from the public? We have 4 minutes still, so if you have any questions, please shoot. No? Are there any urgent questions that the jury still has? Because we do have the jury. Do you have any questions from the judges? Okay. Then if there are no more questions, congrats.
Thank you.
Okay, and now from seagrass, we switch to trouts and essential oils with the presentation from Camila Piran.
We just wait a bit so that the online opens, because we can schedule at 2:30, so that we're sure that the ones that want to join online are still there. So you can warm up a bit if you're not warm enough.
I said they made quite a big change in terms of topics.
You're gonna have to work PowerPoint, and that's not.
Yeah, true.
Your fan as well. Sorry, yeah. Yeah, there's no plug. It's okay. Yeah.
Okay, we did.
So Hi everyone, I'm Isabella and I'm going to talk to you today about my ongoing thesis project because I'm still not yet finished. I've been working on a pathogenesis study of the bacteria Flabobacterium saccharophyllum on the progression and the way to infect it in a lab setting on rainbow trout fingerlings of approximately 1 gram in weight and the potential treatment of this bacteria with essential oils. So why do we care? Why is the species of fish important and to be able to farm them in a good way? Globally, trout is one of the most important economic resources globally. And in Europe, it is the most farmed fish species. The output of the species is used mostly for food consumption, for us, for people, and then the majority of the waste products for that is towards animal feed and to create fertilizers for agricultural use. And they are also grown to stock recreational ponds for sport fishing or just for people to have, or to stock rivers and lakes where they are native to keep the populations. The disease is called either rainbow trout fry syndrome or bacterial cold water disease, and these two differences are primarily because of the geographical location, the age of the fish, or the bacterial isolate that is found. And the main difference that I found in literature is that bacterial cold water disease is mostly the name given to the disease when it is on older fish. They show in clinical signs the same kind of symptoms, which is they start by creating lesions in the skin, and then the bacteria starts eating away at the tissue, and we can observe it in a few various ways. So the first image that I have up there is a very common one, where they attack the back. Well, they don't. They start at the back of the dorsal fin, and then they start eating away. You can also observe it in the eye. You can see in the gills that it has a little mucus and it's swollen, and sometimes it'll have a little yellow pigments. And you can also see in this one picture that the fish's eyes are protruding slightly from the head. That shows swelling. And this has become a very big problem in fish farms because it creates very high mortality on very young fish. So it's a big problem for hatcheries. This disease attacks salmonids, in general, but for rainbow trout it has become a big problem because it aligns very well with the temperatures. So this bacteria is optimal at a range from 10 to 14 degrees, which is the exact temperature where the fingerlings are at when they are in their developmental phase. The bacteria is a gram-negative gliding bacteria, so it doesn't have fimbriae or villi or flagella. It is, and it has become antibiotic resistant, which is one of the biggest challenges that we are having. Oops, sorry. So the current method to treat these outbreaks in fish farms is to go with antibiotics, which is not great because once the bacteria becomes resistant, then we have to increase the dose or change to a different medicine, and then this is also very polluting for the waterways, and just it's not a great situation overall. There has been some research done to see if maybe a vaccine could be a possible way of tackling this issue, but the problems that is encountered is you can either vaccinate the broodstock, so that the mother, before the eggs hatch, and then she passes on some of that immunity to the spawn. Because vaccinating the fry at such a young age, they don't have the developed immunity to take the vaccine yet, and practically it's not very efficient. When you have very small fish and you need to inject every single one, it's not working that well. So this is where the essential oils come in to my project. We know that essential oils have antibiotic properties, and from research I can kind of adapt what has been done before with other bacteria and add it to this bacteria first in vitro and then in vivo to see how this could potentially be an option to be used in fish farms on a larger scale. So from all of this literature review, I came up with the following objectives. So for my thesis, I want to be able to successfully create a challenge with an immersion bath for the fish with the pathogen without creating a man-made port of entry. Because in studies before, what has been done is a fin clip, or they strip the mucus layer to be able to help the bacteria to attach to the host within that bath. But I want to investigate a more natural way that this happens. And at what density I need to make that bath so that I can get this to happen. I want to document the progression of the disease in a lab setting and see what the steps are throughout all of the days. I want to also experiment whether the oils have any effect on the density of the bacteria on the host when I add them with the pathogen immersion. And I want to see if they can, in fact, be potentially used in either transport situations or in fish farms if it makes sense. And this led to my 2 research questions, which is, can they be successfully challenged, and at what density can the bacteria needs to be immersed to reach this challenge? And can do the essential oils basically have an impact or not? So to do this, I designed a series of experiments. The first one is I had 7 strains of the bacteria available to me. 5 were sent by my co-supervisor from the US, and 3 were purchased at the BCCM collection in Belgium. And I performed growth curves from all of them and selected 2 to continue to the in vivo part of the project. The second step was to do a minimum inhibition concentration of the essential oils to the bacteria, and then to test if that concentration that is showing signs of inhibition is not toxic to the fish when I expose them. So for this plate, what I designed was to be able to have my 2 oils that I selected, which is thyme and cinnamon, and I have 3 replicates for each concentration that I made in a 2-fold dilution up until the last 2, which were full dilution down. I have 2 antibiotic controls, also with 2-fold dilution from a standard practice from the CLSI. I have a sterile control, which is just the broth that was used for all of the other wells. Growth control, which is the broth with the bacteria, and an ethanol control. And this ethanol control is necessary because we know that oils and water don't really mix very well. So in order to get that working for a bath, I needed a solvent to get that oil to disperse well. So one of the aspects that I had to work with was at what ratio I had to mix the ethanol to the oil to get this nice mixture where I still have a low enough amount of ethanol that is not toxic to the fish and a high enough amount of EO that will show some effects. So from this control, I want to see that my ethanol is not actually reacting in any way to giving the bacteria any inhibition. And these are the results from that minimum inhibitory concentration. So this is for the thyme essential oil, where we can see that the higher concentrations didn't have any growth. You can see the control in red, where we see a normal growth curve of the bacteria, and then the 2 lowest concentrations, we can see delayed growth and at a lower density. So this shows that the thyme oil is working against to inhibit the bacteria in vitro. For the cinnamon oil, we also only see the first clear growth curve at the lowest concentration. So this is also working to inhibit the bacteria. And my results were as expected. There was no interference with the ethanol, and there was no growth in the antibiotic controls. And from the 2 graphs, I selected the cinnamon oil to continue for the in vivo trial since it had the growth at the lowest concentration, whereas the thyme had at the lowest 2 concentrations. For the toxicity trial, then, I tested out the 2 lowest concentrations of both oils to see how the fish would react. And I did that by immersing them in 5 liters of the same percentage of oil that I did for the in vitro parts, with 11 fish in each replicate, and then they were taken out of this bath into clean, aerated water experimental tanks to recover for 12 days. From this experiment, I also tested a higher concentration to see what the effect would be to reach a toxicity level, and I discovered that at this concentration, so 0.01% of oil in the bath, the fish were showing signs of stress, and they were showing signs very similar to what we would see when you were sedating fish for medical. So I think that this is a very valuable thing to do more research on, because it's a natural sedative that could be kind of like what we use clove oil for, but now we have a broader range, so cinnamon and thyme work in this way. The next experiment that I did was the pathogen bath, which follows the same design. So I created the culture of bacteria to the concentration that I wanted to have in each 5-liter bath, where I kept the fish for 60 minutes, the same as the toxicity trial. I did this by growing them from step to step, so this bacteria is very slow growing, so it takes 4 days until they reach the point in their exponential phase where I need to pass it to a bigger jar. And when I had 3 liters of culture for the 2 strains that I wanted to use, I centrifuged it into small pellets, and then I added that to the water to create 10 to the 7 colony forming units for the 5-liter bath that I exposed the fish to. And this is the baths. From this, this experiment finished a few weeks ago, and I still have to process the samples, but the outcome of this was that at 10 to the 7, we didn't see any clinical signs of infection. So from this information, I go forward with my next experiment, and I know that I have to do a higher density of bacteria if I want to see any effect, if it will work without the portal of entry. So the ongoing experiment that I started last Thursday is everything, all of the treatments that I have brought up into one experiment. So I have 5 replicates per treatment, where I have 16 fish in each bath. I have the essential oil with the strain that I selected from the growth curves. I have just the strain, just the oil, and a negative control that received the same amount of manipulation as the other treatments. So they were all in there for 60 minutes, and then they were taken out and put into the recovery tanks. And the plan for this is to sample for 2 different types of data. So I want to do bacterial reisolation, and I want to do live bacteria plate counting, which you cannot see with qPCR because that also counts the dead bacteria. And this is done by taking swab samples or mucus samples from the fish once they have been euthanized, and having the bacteria grow and then counting again to see the amount of colony forming units that were on the host skin. And with the qPCR, I want to confirm that the bacteria that I'm seeing is the specific strain that I gave them, and not something that might have grown in the stressful situation or just that they already had it themselves. And what I expect to find from this is that if the challenge is successful, I will establish a density, a bacterial concentration that can be used for an emergent challenge where you can have this clinical signs of infection to document and for future research. I can confirm that it is actually my strain that was given and it wasn't conflicting things. I can say that I can see if the addition of the cinnamon oil to the bath impacted the density of the bacteria on the skin of the fish, or if it was the same, or if it made it worse because it made the fish stressed. That's all results that I will have to see. And I will see if it lessens the symptoms, like the behavioral symptoms or the physical symptoms that the fish experiences. And if I think it's a lot already. And if the challenge is not successful, then at least we know that a port of entry wound might be necessary for these kind of studies, and that going in this approach might not be the best way to go for the future. It can be considered that the time of my immersion baths could be modified. Maybe they need to be in the pathogen bath for a longer period. And that the oils don't really work in vivo if it is unsuccessful. And the take-home messages from my thesis is that I now know that these two oils are inhibitory towards the bacteria, at least in a lab setting. I have established a safe range to use these oils for the fish. And if my bacterial challenge is successful, it will be the first one where we manage to infect the fish without a port of entry wound or stripping of the mucus layer on the skin. And for future research, I recommend a wider range of EOs because there's a lot that can be done. There's a lot of plants and a lot of things that we can see. And it would be very interesting to see a comparison between fish that received that wound and fish that didn't, to see if it takes longer, if it is a higher density and more aggressive symptoms, or just anything to be able to compare the two observations.
Which I know that more or less zero, so they're very interesting. Yes, the cinnamon and palm oil is something that the idea to use and to test these types of oil derived from previous experience.
They're mostly from literature and from experiments also done in vitro and one in vivo with a different species of fish. They selected these oils to contain.
Are there any other kinds of plants that have been tested for this that have shown some effective or more interesting from my point of view? Any species of seaweeds from which any kind of compounds have been?
In the case of seaweeds, I'm not 100% sure, but for the plants, there is a very wide range of essential oils that is being used, especially with different bacteria more common, like E. coli or Aeromonas, or other types of bacteria, mostly in vitro, to see also the oils, how they are affecting the bacteria, so the compounds within the oils and how they attack the cells, which is very interesting. And for aquaculture use, I have come across a few studies that use a different plants that I don't know the common names, but it worked. They did an experiment with catfish where they managed to have it lessen the effects of the bacterial infection.
And are this is the kind of oils that are already applied in aquaculture, maybe the
commercially,
aquaculture plants in which they are really rough and to keep the fishes healthy?
Commercially, as common practice, I don't think so yet, but there is a lot of research being done to potentially have this be common practice, because one of the biggest aspects of, like, the promising thing of the oils is that they are volatile and they are biodegradable. So it is a very good thing to use that will not pollute the waterways, because that is one of the biggest problems with agriculture and especially with antibiotics.
Okay, look at I wonder what is your feeling about this? Do you think that this might become the practice of the large use for the?
I think it's very promising. I think it's also very good for preventing it to become an outbreak in stressful situations. So, for example, the 60 minutes that I did the baths, I had in mind in transport, since these are very small fish, you move them from the hatchery to the bigger pond or to whoever bought them. They are placed in these big bags and they are moved from one place to another. So if these oils show an effect to stop the pathogen from attaching properly or to lessen the effects, it can be administered into the bags when they are being transported to prevent.
Okay.
I think that's very
Because,
interesting.
I was wondering about the amount of plants that will be necessary to extract
create
enough
this, yes.
the oils of the ethylene for large scale
Yeah,
applications.
it's very little amounts that is used of the oils. So it is a lot of, yeah, you would have to grow plants and create these oils, but the amount that is being used for treatment, it is very, very little. For the volume of water, it is not that much.
Thank you.
Yes, yes, thank you. It's also it's new for me, but I really like it. I also liked how much you did, and also the follow-up of your research. Like, it's a really good work. I have some questions. So this essential oil is a bit similar, but is it already used in terrestrial systems? Is it used for human health? Or is it something completely new to battle these bacterial infections?
Well, I don't know if as a common, like something that a doctor would prescribe you. I don't think so, but I know that there is a lot of like home remedies and like homeopathic medicine that goes in this direction.
Yeah.
But and I'm not sure for terrestrial animals. I know that there's a lot of research being done, including these oils in the feed, and testing how that reacts and how the animal assimilates it in that way. But I didn't go in that direction.
Here in Italy, we use the thyme for cooking, because it's spices.
Yeah. So, yes, a bit about how does this work in the physiology lab? How are these 7 oils? It's not too much illegal, but just what is bacteria, right? You showed the graph.
Yeah, so I have, this is from a study that I cited in my literature review. Sorry, it's a little far back.
Yes.
This one, it mostly attacks the cell wall of the bacteria. So it's these are like the main 4 nodes that it attacks. So it'll like, oh, sorry, it breaks the outer membrane going through, and then it can also attack the organelles within the cell. That's like the main mode of how it works. But for my work, it's more the experimental parts, and then
So you say that to put it in water, you need ethanol.
yeah, I'm using ethanol.
With these fishes, fish experiment, you also put ethanol in the water.
Yes, I did.
So did you have a control for ethanol?
Yes, in the toxicity, when I did the toxicity experiments,
Yes.
that I had my 2 oils, I had an ethanol control with the same concentration with the
Yeah.
fish.
Okay. So it's really an essential oil and not
Yeah,
ethanol.
so I did both in the lab on the MIC, and I did with the fish.
Perfect. Do you think if we apply this essential oil, there can be an essential oil resistance?
Yes, that's a really good point actually, because that is one of the points for choosing which oil to use.
Yeah.
Because if they are fully inhibitory, that can lead to the bacteria developing antibiotic resistance. So the 2 concentrations that I use are actually these 2, because I think it's also interesting to see how they're affecting the virulence of the bacteria rather than inhibiting the growth. And I have 2 concentrations for my in vivo trials, and then I can observe at the lower concentration where it's not really inhibiting the growth but delaying it, if it's different to the one with the whole.
And one more question. So, if this works, is an essential oil, is it an antibacterial compound, or how should I see this?
They have antibacterial compounds, so the essential oils, I think they're called terpenes, the compounds in the oil themselves, that have antibacterial properties that attack the cells.
Yes. Okay. Thank you.
Yeah, thank you for the presentation. Again, I'm not an expert of the feed, but I was just wondering if these oils that you use, they float. I think they float somehow. They in the column of water.
Ah yes, but that is why I
You
added
use
the
that?
ethanol
Yes.
to be able to. I went through a very long lab phase where I was testing different types of solvents, keeping in mind what was more eco-friendly, because for this, DMSO has been used a lot in the past, but that's very toxic. So to add that into waterways is not advisable, so I wouldn't really want to go in that path. And then with the ethanol, I found out that if I mix with a 1:50 ratio of essential oil to ethanol, I can get it to disperse well in water, and I waited a few days even to see if it separated or not.
So you check if your solution was homogeneous or...
Yeah, exactly. So I went through that process.
And do you think that can be also, this strategy can also work in the field, like if you add some fish farms?
That's the goal. If this works, then we should
in the
look
large
into
scale,
how to make this into a bigger scale, because this is a very eco-friendly way to treat like aquaculture animals, adding this to the water itself in the farm.
Thank you.
You're welcome.
Okay, so we still have some time, so if the public has some questions, you want to ask? Then I might have a question, actually, there's a follow-up question, because it would be indeed interesting to introduce them in the field, but if they are antibacterial, then what about the bacteria in the sea that are actually part of the ecosystem? How do you then differentiate between the bacteria that are pathogens and the bacteria that are actually key components of the ecosystem?
That's a very good point. I think more research would have to be done. Obviously,
It's
but
always
I
the
think,
problem with
yeah,
going in the field.
I want to say that because these essential oils are so volatile, that they biodegrade very quickly. So I think for it to actually like become bad for the bacteria already in the water, it would have to be used a lot.
So you think once they introduce them, they would be local, like they would stay and they wouldn't be transported because their incubation time is shorter.
It's
than
very
the
short.
usual.
And even when, like in the lab when I was pipeetting, I had to be very quick because it's very volatile.
Okay. Then maybe. Okay,
We have time for one question, just a very simple.
yes, yeah.
So it's related. So this fish
Yes.
and bacteria. The problem is they grow at the same temperature in the aquaculture ponds. So that
They
is why.
coincide, yes.
Why not just let's say you grow the fish at 9 degrees or 8
Well, they
degrees?
thrive at 10 to 14, but you can find this bacteria at lower temperatures.
Okay.
That's it's the optimal growth is 10 to 14.
Yeah.
Yeah. And also, like for the trouts, you like as the seasons get warmer, you want to increase the temperature to also help the development of the fish and everything. So.
Okay. No more questions. Thank you very much.
Then you can have a bit of berries.
I appreciate that.
Yeah, plates.
Not when I talk.
He's the...
Yeah, that's circle now. Okay, we're set up. Very good. Recommend.
Okay, good. So from seagrass, from trouts and essential oils, we go back to seagrass systems with the presentation by Joel. You can start.
Thank you. So, hello and welcome everybody. My name is Lo Larsen, and this is my thesis defense on the role of chemosymbiotic bivalves in the nitrogen cycle of tropical seagrass ecosystems. So, as I assume most of you are familiar with now, seagrass ecosystems are very important. They provide a plethora of different benefits for us, such as carbon sequestration, they produce a lot of oxygen, and they are also crucial habitats for a lot of marine species. However, recent decades have seen worrisome decline in a lot of these habitats all over the world. This is mainly driven by anthropogenic factors such as climate change, coastal development, pollution, etc. However, recent studies have found that when there are chemosymbiotic bivalves living within the sediment of seagrasses, they can actually be more resistant to some of these stressors. One of these bivalves is Codacea orbicularis. It's a part of the Lucinidae family, which is the largest family of chemosymbiotic species. They have a symbiotic relationship with sulfur oxidizing and nitrogen fixing bacteria. So they have specialized gills, cells within their gills where they host the bacteria, and the bacteria can use sulfide within the sediment to chemoautotrophically produce organic carbons. These clams are found in the shallow waters of the subtropical, tropical Western Atlantic, so all the way from Florida down to Brazil. And they bury into the rhizosphere of the seagrass Thalassia testudinum, more commonly known as turtle grass, which is common all over the Caribbean. So to understand the system, you can look at this figure right here. So as you see, in the water column, you have high oxygen and low sulfide. But the further into the sediment you go, you usually get higher and higher concentrations of sulfide and lower concentrations of oxygen due to metabolic processes of microbiome. The clam then positions itself at the perfect place within this gradient so it can access both oxygen for its own metabolism, but its symbiont can access sulfide for its own metabolism. So, obviously I want to research the system because seagrass beds are important, as you've all heard today. But a lot of the studies on why these clams are important are quite contradictory, especially when it comes to their role in nitrogen cycling. It's also a very hard system to study due to the fact that we have this environmental gradient and that oxygen and sulfides are very reactive with each other. So the goal was to develop a new system that allows for the accurate measurement and quantification of Codacea orbicularis' impact on its environment, specifically looking at nitrogen cycling. To do this, we developed three main hypotheses. Number one is that Codacea orbicularis serves as a net source of ammonia in its environment, stemming from nitrogen fixation pathways. So as I previously mentioned, the symbiont of Codacea orbicularis has the genetic potential to take dissolved dinitrogen gas in water and produce ammonia. Hypothesis 2 would be that both ammonia and nitrate can be utilized as a nitrogen source for the Codacea orbicularis symbionts. So once again, even though they can fix dinitrogen gas, studies also find that they have the genetic potential to take up nitrate and ammonia from the water, and also use nitrate as an electron acceptor during redoxing. And the third and final hypothesis is that the gill symbionts are the primary site of nitrogen uptake, with subsequent translocation to other tissues. So basically, we're expecting that within the gills, where the symbionts live, is where we're going to see the uptake, and then as the clam either eats or milks its symbionts for nutrients and carbon, this will then be translocated to tissue without symbionts in them. So to do this, we had to engineer and design a new system. We wanted to mimic this natural gradient that you find in nature. To do this, we had a two-inflow system where we could push water in at the bottom of the sediment containing high concentrations of sulfide and nutrients, and just above the sediment we circled oxygenated water so they had access to both of these parts of the gradient. We also had complete control over the nitrogen we put in, the amount of ammonia and nitrate, and we also used stable isotopes to be able to track the fate of these nutrients within the system and within the clams. The clam then self-positions within the gradient just like it does in nature, and it allows for these very biologically relevant experiments. We engineered 8 of these chambers, and we then split them up into 2 main treatments. We have treatment 1, which is isotopically labeled ammonia, and treatment 2, which is isotopically labeled nitrate. I don't know how many people are familiar with isotopes here, but if you're not, just think about it as a little flag. So we will know, in this treatment, we will know where the ammonia goes, and in this treatment, we will know where the nitrate goes. So we ran the system for 48 hours to create kind of some equilibrium. And then after this we sampled before we put any clams in. Following this, we took samples every 24 to 48 hours of effluent nitrogen, so all the ammonia and nitrate leaving the system. We did oxygen measurements to make sure that the clams are alive, because you can't see them because they burrow. We also want to see if we could measure respiration rates, as well as pore water sulfide, which we took from the bottom to make sure that we were creating this pool of sulfide we wanted at the bottom. At the end of our week incubation, we took the clams out, dissected them, we took mantle, foot, and gill tissue, and then we sent this out for isotope sampling in the lab. And we also did some morphometrics to be able to normalize our results for the size of the clams. Suspense. So, what were our findings? Well, if we first look at the F1 of the system, so this is ammonia and nitrate leaving the system. You can see at time point 0, before any clams are in the system, we are higher up than any subsequent time points in time points. We're expecting this time point to be the same as the dotted line, because this is our calculated baseline. If everything has gone completely smooth, this is where we would have been. Does not really matter, because what we see is that once the clams are added, immediately after time point 0, we see a drastic decrease of nutrients leaving the system, especially for ammonia. This is obviously statistically significant, and here that doesn't happen until here, but still it remains low in both our treatments, or in all 8 of our chambers. This is further supported by our isotope data that we collected. So, as you can see here, in our heavy ammonia treatment, so this is when we have a flag on our ammonia, we see that especially in the gill, but also in the foot and the mantle, we have high assimilations of these flags in the tissue. And what we're comparing with here is untreated natural abundance. So these are clams that have just been living in an aquarium and have never been exposed to any heavy isotopes. So they are what you would call biologically relevant, just baseline for heavy isotopes in the tissue. We do not see the same trend for nitrate. As you can see over here, nitrate is fairly close. These natural abundance are the same, it's just that the y-axis on these two graphs are drastically different because we see uptake on this, but very, very little on this. And this is, even though you can see a little bit of an elevation on nitrate, it's not statistically significant, and we can't say with certainty that they actually utilize nitrate. We also see some evidence of translocation. So remember that I said that we expect the uptake to happen in the gills, the darker area here? That is what we see, because we have far higher assimilation in the gills compared to any other tissue. We then expected to see that this uptake would then be translocated to other tissues that are aposymbiotic with no symbionts, and we see that exact thing, that we have higher concentrations in both the foot and the mantle of our treated clams than we do in non-treated clams. So there is likely translocation, it just takes longer because the clam has a slower metabolism compared to bacteria and so on. And we also normalized this for the weight and the time, and calculated the assimilation rates of nitrogen for 2 different treatments. And as we can see for ammonia, which you have right here, our results are fairly similar to that of other chemosymbiotic clams that has been conducted in the 1990s. You can also compare it to Mytilus edulus, which I think most of you are familiar with as the common blue mussel. This is an aposymbiotic clam with no symbionts, and the uptake here is far, far lower, an order of magnitude lower than the symbiotic ones. So we're obviously seeing uptake through the symbionts. What does all of this mean? Well, a lot of previous studies find support for excretion of ammonia. So they see that if you look at the genome, if you look at transcriptomics, even if you look in the field or in the lab, you can see that they should be and they are excreting more ammonia than they're taking up. However, this study finds the complete opposite. We see that we're removing a whole bunch of ammonia from the system and that it's being assimilated into the clam and into the symbionts. We're not the first to see this. There has been speculations previously on other leucinid clams, but also on the same species, where they see that in nature, they're actually associated with lower ammonia in their near vicinity, but also in mesocosm experiments that they take up a lot of ammonia. Another thing we measured was oxygen, as previously mentioned. We ran some controls, and I would have controls for everything, but we haven't gotten all the data back. But we have the oxygen controls ready. And as you can see here, that for the abiotic control, which is the exact same method, just no clam in there, it's just sand, and for the inoculated shell control, which is just dead clam shells that have been dropped in an aquarium with live clams to make sure they have the same microbiome as a live clam, you can see that we have very, very stable oxygen consumption. And we have more oxygen consumption in the inoculated shell control because we are introducing more bacteria. When we look at the treatment, where we have live clams in, we have a far higher variance, both within the group, which is the shading here, but also between days. This is likely because the clams are active. They are digging through the mud, they are pumping water up and down. They also, if we stress them out, they will close up and become essentially metabolically inactive. This kind of things. And using this data, we managed to actually calculate the respiration rate of Codacea orbicularis. So right here. There's been very, very limited work on respiration rate on lucynid clams in general. This is the second study to do so, and we're the first one to quantify it in this way. So we had to kind of rely on previous studies of other clams to see if our results are reasonable and if the system works as intended. But as you can see here, we are fairly close in oxygen uptake rate to a lot of other studies. A common theme you can see is that we have a lot of variance, and this is common for bivalves because they, depending on temperature or stressors, they close up, they open up, they move around. It's pretty hard to have a consistent rate. But we are reasonably close. And finally, we also looked at sulfide, right? So what you can see here is that at time point 0, before the clams are inserted, we start off pretty low. We wanted to stay around about 80 micromolar, but as soon as the clams are put in the system, and the next time we sample, we are where we want to be, and we remain stable throughout the experiment. So this is, again, we managed to create this pool of sulfide that we needed to create the natural gradient seen in nature. So the oxygen results and the sulfide results together tell us a story about the fact that the system we engineered works well. We're seeing that there is enough sulfide at the bottom throughout the experiment, that it's environmentally relevant, and they're also having enough access to oxygen that we can see fluctuations. They can be metabolically active as intended, and the system seems to work just the way we want it to do. So, what can we conclude from this? Well, first and foremost, contradicting our first hypothesis, Codacea orbicularis seems to be a net sink of ammonia in the system. So even though a lot of other people have speculated that they should let out more ammonia, and especially more than other non-symbiotic bivalves, we see the complete opposite. And there's a few caveats to this that you should keep in mind. So this clam was exposed to starvation, more or less, because we feed them, the water we provided the system was artificial seawater. So there was nothing edible in here, and this is a mixotrophic species. They can both rely on their symbiont, but they can also filter feed. So in the conditions we provided, they are purely relying on chemoautotrophy and their symbiont. And their interaction with the nitrogen cycle would obviously be different if they had access to eat other things. We also see kind of a contradiction to a lot of previous studies, because a lot of previous studies say, well, these clams are benefiting seagrass beds, and seagrass beds really, really love ammonia. So that's probably one of the drivers to why it would be very beneficial for this. But we propose that there must be some other factor that is the main driver of this, the fact that they're very beneficial for seagrass beds. And it could be that they're removing sulfide at high levels or other environmental factors. For our second conclusion, we see a significant uptake of isotopically labeled ammonia in all tissues, so we're partly confirming our second hypothesis. We see very substantial evidence for the fact that they prefer ammonia and they take it up. We do not find this for nitrates, so we can only partly confirm this hypothesis. For our third hypothesis, we see that translocation of symbiont-mediated ammonia happens in symbiotic tissues. So the fact that we have the highest uptake in the gills indicates that we have that the uptake happens through the symbiont and not from the clam. And especially if you compare with non-symbiotic clams, you see a similar trend. So everything's pointing towards the fact that the symbiont can take up ammonia, and the clam can then eat its symbiont or milk its symbiont for nutrients and carbons. And finally, this was the first study to quantify the oxygen uptake rate of Codacea orbicularis, and it was similar to that of other bivalves. So all in all, the new system was a success, and we're hoping that this will kind of lay the baseline for a lot of future studies utilizing this kind of novel two-phase system where you can look at chemosymbiotic animals or any species generally under long periods of time without having these gradients, evaporate from abiotic interactions. And I would like to thank Ian Hughes and Professor Peter Gerges for all of their help. This project would not have been possible without them, and also the organizations and foundations that have supported me financially through this. Thank you so much, and thank you guys for listening.
Thank you very much, Hello. And yeah, during
this is the organism with which I'm not familiar, but okay, it's a chemosymbiosis, but I mean, it will be the symbionts, here,
I mean, who's the symbiont?
yeah, okay, I mean, the bacteria that live in the,
Yes.
okay.
Yeah. So they're a free-living type of bacteria that's found in seagrass beds all over the planet. But we see that they can colonize host gills that are kind of specialized for this cause.
Okay.
And within them, they get this perfect condition where they can live on, but the host still eats them. There's been pretty conclusive evidence for the fact that the host eats them, so
Okay.
it's a symbiosis in that sense.
So, I mean, but there is a Podacia ornicularis able to survive without this bacteria.
Yes, there has been a few starvation experiments on a lot of different designers, and what they see is you can make them lose all their symbionts. It takes months and months of starving them because they slowly consume them. They really want to hold on to them. However, as soon as you put them back in their natural environment, even just for 24 hours and bring them back, they will reacquire them. Yeah, because they're very important for their metabolism.
Okay, so what you call aposymbiotic tissues will be tissues in which these bacteria do not occur, right?
Exactly. Yeah. Yeah.
Okay, well, this is very interesting. Okay, then do you think this may actually, I mean, the behavior like this may actually cause a damage to the seagrass? I mean, this may be a form of a competition for the nutrients.
That's a great question, and that's something we're also, we're not done with this, obviously. We haven't even gotten the controls back for the nutrients, but we have more experiments lined up already, because that's the hypothesis that we've proposed now, but there's a lot of science pointing in the different direction. So we really need to verify our findings now first, and then look, can we see, if we label dinitrogen gas, can we see uptake of this? And if we feed them, can we see high excretion of ammonia? And then we can more say, during kind of biologically relevant conditions, where they have access to filter feeding, at least at times, will they be a net source or a net sink? And then I guess we could really conclude, are they competing or are they helping when it comes to nitrogen cycle?
Maybe any chance that, okay, in different times or different phases, they may behave
Yeah.
different.
Definitely, and also with tidal forcings and things like this, the sulfite concentration may vary, and during these times, I mean, they can't rely on chemoautotrophy anymore, and it might be completely different. It's a very complicated system to study, and there's a lot of experiments needed if you really want to pinpoint how they interact overall at different times and different conditions.
Okay, and then there's a technical question. Okay, you created this system okay, in the laboratory. You have a you create a nursery.
For the clams?
Yes, I mean these chambers.
Yeah, so the clams, before I arrived, they went down to Florida and they collected the clams and they just have them living.
Okay, I mean these chambers. Okay, physically, I mean.
Yeah, we built them from scratch. Yeah, designed them and built them. We tried them.
You have them in the laboratory.
Yep.
Okay.
Exactly. Yes.
Okay, now looks that obviously there was a very careful thinking, very good planning. I don't know, I wonder if really, from this point of view, these chambers can perfectly recreate the conditions in the field.
That's a great question, and we have some more experiments coming where we're going to try and set up the system just like this without a clam. And then if you can flash freeze them, you could then cut them in sections, and that way you could actually quantify exactly what conditions are throughout them. But just the fact that we know that down here you have high sulfite and no oxygen, more or less, and up here we have high oxygen, especially when the clams are metabolically inactive, means that somewhere in the middle there has to be a gradient. And the clam doesn't need a perfect gradient because the clam will find it on its own. That's the best thing about it. So you just need to know that you have two different pools, and then the clam will do the rest.
No, I wonder if the person who supervised it for this project have any plans to try to recreate something. I mean, do some kind of similar experiments, but directly in the field, is that technically feasible?
Yeah, I guess you could, and there are people that do that. Other people, the scientific field is quite small, but I know there are other people that do that. It's very hard because there's just too many parameters that are coming into play. So that's kind of the reason why we did this, was because it's very hard to. People get such different results in the field because there's too many parameters. So we're trying to really isolate what we can control and see what each parameter does.
Okay, one final questions. Actually, in the field, do those that have natural predators, anybody who goes there and prays them?
Yeah. So one of the main reasons why Kodakia is so happy with the seagrass is because within the rhizosphere, they're safe from eagle rays, and eagle rays is one of their main predators, so they can't really dig them up when they're deep in the rhizosphere. Thank you.
Yes, thank you. It's been very interesting. Very good presentation, very nice study. So these bivalves, are they common or present in all bioregional seagrass meadows? Or it's specific for that bioregion, or you can find them in the Mediterranean, you can find them in the Pacific as well. The same species, but I mean,
Yeah.
same bivalves and symbiotic relationship.
Yes, the Lucinidae family is omnipresent all over the globe, in every ocean.
Okay.
And you find them even in the deep sea at certain places. You can find species.
Yes.
Yeah, so they're all over.
So they are not restricted to seagrass beds.
Well, not all of them, but they are generally associated with seagrass bed, and they generally specialize quite a lot on a specific type of seagrass. This one, all studies point towards that you only find it in Thalassia testudinum, but I've been diving in a bunch of seagrass beds where you have like Thalassia testudinum right here, and then you have another species, just slightly different. And maybe if someone were to dredge or really look for them, they might colonize some other seagrasses.
So there are specific species which only appear in seagrass beds?
Yes,
Okay,
yes.
that's nice. Yeah. So you say that these clams, has there already been studies done on the positive and negative effect of these clams on seagrass? Did they remove these clams and then saw the effect? Or?
Yeah, the only studies that have been done indicate that they're positive for the. Yeah. And no one has really pinpointed why, but they see particularly if you have a lot of turbidity in the water or a lot of heat stress, then the seagrass survives better than if not.
Yes.
Yeah. There's pretty good studies on this, very elegant.
Yeah. Yes. So you say, based on your results, you say there's a negative effect on seagrasses. Can you explain why? Maybe make that.
I don't think there's a negative effect on seagrasses, but I do think that there is potentially a competition for ammonia with seagrasses, which is something that no one has really thought, because people think quite the opposite, that they're providing a lot of ammonia for the seagrass.
And if they have competition with nitrogen for seagrasses, you will also have competition with algae, and can it not be that because of that they have a positive effect on seagrass meadows because the negative effect is.
It could be, but I'm not an algae expert. But how I would assume the system looks is that you have the dissolved ammonia in kind of the top layer in the water column that benefits the algae, whereas they mainly affect their microsphere where they interact with ammonia in the sediment and they release ammonia in the sediments. So.
And what is your next phase, your next experiment?
So the next one after this is, well, hopefully our controls look good, and if they do, then we will go over to, my supervisor has already started doing mixotrophy to see how this changes when you start feeding them. And the next one after that I want to do is with labeled dinitrogen gas to see if they're actually taking it. And then after that we're going to have to see what the results lead us.
Okay. Thank you.
Thank you.
Okay. Thank you for the nice presentation. Again, I'm not an expert also on the field, but yeah, you show some graph where there was the concentration of the system of the nitrogen, right? Just a curiosity. So in the right graph, so at you have an increase of I was wondering how which, so this is the concentration of nitrogen in the water, right, of your system.
Yes, yes.
So there was the production of. So did you add some other?
We had, so the dotted line is
So
how much we.
Okay.
How
So
much-
the first, sorry, at the time zero is the initial concentration, right?
Yes.
Okay.
Yes. So this one we imagine would be exactly on this line. If everything was perfect, if sampling was perfect, if the pumps were perfect, this is where we would have been. And then even though we're pumping in from two different ways, everything is let out through the same. So we will know exactly how much is disappearing or being added, because we know exactly how much we're putting in already.
Okay.
Does the public have any questions? Can I ask a question? Yeah, go for it. Yeah.
Yeah, it's a very interesting study, definitely. I'm very curious, because you said we found that there was a net uptake from ammonia, but a lot of literature says that there's a net release of ammonia. Why is there this difference? Was it an assumption that was made, or are these also experimental results?
Well, they're all kind of experimental in the way that there's no one. What we think is that no one has really figured out how to do this in a good way. Because there's been a lot of different tries in the field and in mesocosm and things like this, but it's very hard to do longer studies where you keep the gradient going and still be able to measure everything properly. But I personally think that their impact on the nitrogen cycle is dependent on their feeding regime at the time. So I think that if you have them in their natural environment and you're feeding them, or they have access to heterotrophy, there will be net excretion, because they will obviously get a lot more nitrogen from eating things. So I think it's mainly during starvation that they are sink. But that's just, we have nothing to prove that yet.
No. Thank you.
I actually have a, not similar, but that is associated to this as a ecologist. So this is a single species experiment, right? Because I understand that of course you want to focus on that. But then have you sampled to see what's actually the macrobiotic community in these seagrasses? What are the other species present? Are they competing also because other macrobiotic communities?
Yes.
Are there other so just if you have a community view on this, or if it's just single species that you're actually looking at?
Well, we haven't sampled. So when we go in the field, we don't want to dredge, and it's super hard to get any type of permits in the Caribbean overall. But you have to dig them out. So you just shove your hand in there, you look for them. So sadly, we've encountered or encountered some of the less fortunate species living in. No, you just go with your hand and you pick them out. And sometimes you get a plant, and sometimes you get a fireworm, and then you're unhappy. So there's
I would be happy.
Yeah. Well, it hurts for the rest of the day, but you could probably look at it at a community level and see a lot of interesting things. But I guess I'm not a ecologist, but I would assume that there are very few other species like nematodes and stuff that would uptake any type of ammonia, right? They would mainly be excretion.
No, yes, of course, but the whole community affects also the material community.
Right,
and
of
so
course.
what I'm thinking is if there's a different community in different areas, then maybe that also happens. And then also, my other question is the comparison between the studies. Have you checked if your methodology is more similar to the ones that have the same results as you? Or because experiments, everyone approaches it in a different way, and you also highlighted it.
Yeah.
So is the methodology that you use more similar to the ones that have the same results, or is it all a mess?
It's a bit of a mess, but I should say that the actual in vivo experiments that they've done in the lab, they are fairly similar to the previous people. Most of them have done some kind of mesocosm setup, and sometimes in really big chambers to mitigate the interaction, the purely abiotic interactions. But no one else has done it with this kind of pseudo flow through respirometry kind of system before, so it's kind of hard to compare that.
Okay, thank you very much.
Thank you.
And I think there's a question from the chat. Okay, so this was the final presentation. Thank you all for
Thank you.
Thank you to the public and the students, and thank you to our jury.
It was fun.
Great.
Just 2 more.
When you are at 13 minutes, I will just put 2 minutes left.
2 minutes left, all right.
Okay.
Okay, you may start.
Yes, good afternoon everyone. Today I will present you my master thesis work, which broadly is about population connectivity in Antarctica. But before going into details, I would like to show you this map because it reminds us something simple but often forgotten. The world is mostly water. The oceans do not divide us, they connect everything. And at the center of this global ocean system, we find Antarctica surrounded by the Southern Ocean. And this is exactly where I want to take you today. So the Southern Ocean is home to the strongest and longest current in the world, the Antarctic Circumpolar Current, which is flowing clockwise around the continent and isolates this region from the rest of the world. But its isolation does obviously not protect this extreme and wonderful environment from a more global phenomenon we all know, climate change. And among Antarctica, there is one area particularly affected by the changing conditions, the Antarctic Peninsula, situated just here. More specifically, the western coast, so the western Antarctic Peninsula, is one of the fastest changing and warming regions on Earth. And on top of that, it's also shaped by complex oceanography, as you can see here, with, for example, the Antarctic Peninsula coastal current flowing along the coast, and several oceanographic features like eddies or deep throat. And in this complex and rapidly changing region, there is one aspect of marine populations that has often been overlooked. It's their connectivity. By connectivity, I mean the exchange of individuals or genes across space. And understanding this is really crucial because connectivity is not only about movement, it also shapes fundamental processes like population dynamics, population structure, or genetic diversity, which ultimately determines a species' capacity to adapt or to respond to environmental changing conditions. So, to understand the connectivity in this region, we needed a good model species. And in Antarctic ecology, one of the best is the Antarctic sea urchin, Stercinus nevaeri. First, because it's one of the most abundant macrobenthic invertebrates, it really structures benthic communities on the Antarctic seafloor, and also because of its life cycle. In fact, it is a broadcast spawner, which means that to reproduce, it will release its gametes freely into the water column. And these gametes will become tiny larvae that will flow and be transported in the water during 4 months. And given this quite long pelagic larval duration of 4 months, we expect this species to be highly connected over large spatial scales. And in fact, this large-scale connectivity has been observed and proven in East Antarctica from a research that was conducted a few years ago. But surprisingly, the researchers also found unexpected genetic differentiation over smaller spatial scales. So what about the Western Antarctic Peninsula? Can we find similar patterns in this region? It has never been investigated for this species. So this is why I study how does the connectivity in the Antarctic sea urchin vary across large, intermediate and small spatial scales along the complex and changing coastal system of the Western Antarctic Peninsula. So, to answer this question, a hierarchical sampling design was adopted with 2 broad regions, so the north and the south, 6 locations, and within each location, between 1 to 3 sites were sampled, so leading to a total of 13 sampling sites and more than 250 sea urchins collected. And to understand their connectivity patterns and their gene flow, we looked at the genetic differences, focusing on single nucleotide polymorphisms, also called SNPs. So what are SNPs? It's just basically one single nucleotide variation in the DNA sequence, and they are highly abundant in the genome. They are also highly variable, so highly informative, and reproducible, so easy to sequence. So they are ideal genetic markers for population genetic studies as this one. Obviously, it's not possible to sequence the entire genome. It's way too expensive. So in our case, we applied restriction site-associated DNA sequencing, which will use a restriction enzyme to cut the DNA at specific part of the genome. So every time it sees that recognition sequence, the enzyme will cut the DNA, and we will end up with short and uniform fragments for each of our individuals. These fragments were amplified and sequenced, and we ended up with a fragment or reads for all our individuals, which were cleaned, filtered, and analyzed through the bioinformatic program stacks, which is really designed for rat sequencing data, to finally build this kind of genotype table where we have all our individuals, all our SNPs, and for each of them the genotype. So this really formed the basis of our analysis, and in our case we ended up with 877 SNPs across 233 individuals, and based on that we analyzed both the population structure through a principal component analysis, or PCA, and an admixture model, and we also looked at the genetic differentiation across our 3 special scales. Thanks to an analysis of molecular variance, also called ANOVA, a pairwise FST measure between our sampling points, and mental tests for isolation by distance. So let's dive into the most interesting part, the results. Our results at large special scale were quite straightforward, because when we look at our PCA, we see really a wide distribution of our individuals, and we don't see any clustering based on sampling location or region. And our admixture model also confirmed this large-scale connectivity, because the model gave best accuracy for k equal to 1, which means that all our individuals are likely coming from the same ancestral population and do not form any genetic cluster based on their region or on the location. And even if we look at higher k values, so assuming that we have several ancestral populations, we see an apparent genetic admixture with the ancestry proportion, which is quite similar in all our regions, in both our regions. This was indeed further confirmed by our analysis of molecular variance, which revealed low and not significant genetic differentiation attributed to differences between the north and the south, and also by our mental test, because the mental test was not significant. So we clearly saw there is no isolation by distance. So here on the graph we see the genetic distance, the geographic distance. Well, we see a weak positive trend, but the mental test was not significant. So it means that the gene flow is not restricted by distance along the Western Antarctic Peninsula. So, based on those results, we can conclude that we have one single, well-connected population of Sterichinus numeri at a large spatial scale, which is, in fact, consistent with the 4-month pelagic larval phase of the species and the Antarctic circumpolar current, which is flowing around the continent and likely promoting dispersal over large distances. But the story doesn't end here, because when we looked at our connectivity patterns at intermediate spatial scales, so basically between our locations, we saw that a significant proportion of the genetic variance could be attributed to difference between our locations. So it was very low, but still significant. And also our pairwise FST comparisons between our locations also revealed some comparisons to be low but significant. So we clearly had signals of genetic differentiation at these intermediate spatial scales. To understand those signals, we looked at the oceanography of the region, and namely the flow of the Antarctic Peninsula coastal currents. And I will use two examples to illustrate the influence of this current probably on the connectivity. So first, if we look at Deception Island, which is located here, it's located within a cyclonic circulation around this island, which we can expect to promote a certain retention area here and likely limits dispersal and gene flow with southern location. And in fact, when we look at our pairwise FST values, we saw that most of them were significant with all other locations compared with Deception Island. Another example, here in the middle, we have three sampling locations right here. Between them, there were not much differentiated with each other, but all of them were genetically different from the Mine Island based on the FST values. So we can also relate that probably to the coastal current, which is like splitting just here and creating opposite directions flows, and this can likely limit the dispersal between those two areas. Yes. So we also looked at the genetic differentiation of connectivity at the finest spatial level. So basically between the sites coming from one same location, so those are sites coming from one unique location. And we saw that between sites, analysis of molecular variants again revealed low but significant proportion of genetic variance, so attributed to difference between the sites. However, when we looked at our pairwise FST values, they were only significant between sites from one location, so the sites within other locations were not differentiated with each other. So this was a bit contradictory. We concluded that fine-scale differentiation along the Western Antarctic Peninsula is possible, but not obvious regarding those results. But we might have reasons that this can be that this fine-scale genetic structuring can be present, because indeed in East Antarctica the authors they highlighted the fact that the species was largely self-recruiting, which means that larvae produced at one point tend to settle actually close to the place where they were born and did not travel that much over large distances. And they also found out that the larvae, because they used larval samples, which we did not, were collectively dispersing, so they were spawned at the same place and then traveling maybe collectively and settling at another place, which can promote that kind of fine-scale genetic structuring. Yes. So I think that our study adds to the growing body of evidence that connectivity in marine species, even with a high dispersion potential, is not uniform, it's not homogeneous. It shifts with currents, with coastal features, but also with the biological traits of the species and with obviously the scale at which we observe it. And as climate change is accelerating rapidly, it's really important to understand how a species can or cannot move and how much they exchange genes to better understand their resilience and guide, I think, conservation efforts in the future. And then I also do believe that the future of Antarctic ecosystem regarding connectivity depends on more than just studying genetics or oceanography separately, but it requires that we bring them together, and future research should work on that, on combining genetics data with oceanographic modeling to really get a full picture of the connectivity in this region and also in other regions of the world. So because as you know, what happens at the ends of Earth does not stay there, and what happens there also impacts us. So protecting those connections means also protecting the planet we all share. Thank you. for your attention.
Thank you very much. So I think maybe ladies first. Marina, we want to start.
Well, very nice. I don't understand those genetics you did, so I have some more general questions about your work.
Yeah, sure.
First of all, at what phase of this 4-month larval period were these samples taken?
Sorry?
At what phase of the 4-month larval period were the samples
Ah,
taken?
yes, sorry, I was not clear, but I didn't do the sampling, but they collected adult specimen. So we just look at the genetics in the adults.
Okay.
But we hypothesized that based on the 4 months larval pelagic phase of the species, we would expect a large-scale connectivity and that they could travel long distances.
Okay, so 4 months of larval phase, that current is really very strong. You can see it in the radar of the ship, so it's a very, really important barrier. But then inside, you have local phenomena of circulation that change a lot. So when you show that map and showed that there's an island and there's a certain, a country current around the island and so on, that probably happens in time in a very short time. It changes a lot. It's a very dynamic system, no?
Yeah, you mean close to the coasts?
The other one. So you explain the real connectivity, so the samples that are closer to each other are more similar based on this local circulation phenomenon. My question is, are they around or they're just very short time phenomena so that in a 4-month larval period they cannot justify this differentiation? You think it's okay to use that interpretation?
Well, I think the Antarctic circumpolar current is very strong, but it's also like not along the coast and it's way further.
That's why, so.
But so
inside, what happens?
So you are talking about the coastal current.
All the other ones, many bays that are in.
I think it's really difficult.
Islands,
Yeah, I think it's really difficult to maybe generalize something that is like so complex and so changing along the entire coast, which is like more than 1,000 kilometers long. So yeah, probably there are like specific oceanographic processes happening everywhere that could or could not promote that larval dispersal and explain the connectivity or the genetic differentiation we observed. But I do believe that this current and the oceanography of the region can in certain cases limit dispersal, even though it's very strong.
All right, you mentioned that you started your presentation mentioning climate change. How do you expect climate change to affect connectivity of the populations in the country?
Yeah, introduced it with climate change because I think I don't expect climate change to affect connectivity, but I think that studying connectivity is important to better understand the resilience and the adaptive potential of a species in the face of climate change, because it really depends on many factors. But regarding the species, as it's like releasing millions of gametes freely into the water column, they will disperse probably everywhere, and the large-scale connectivity that we expected, we have observed it. And this dispersal will also facilitate and promote like exchange of genes, and it will enhance their gene pools, and there is more chance that this kind of species probably will have the trait that is necessary maybe to adapt in a changing environment. But it really requires more more specific analysis to see, because the single nucleotide polymorphisms, they can be neutral or they can be adaptive, and we would need to investigate if we use like adaptive markers or not to see if it was like related somehow to the changing conditions or not. But it's more about like understanding how they are connected, how much they exchange genes, and also their ability to move also to different places, probably southern if the climate change is coming from the north, even though we are already quite at the south end of the planet. But their ability to move and to exchange genes to enhance their adaptive potential and the ability. Yeah, that's
Okay, thank you.
thank you.
Thanks for a really nice study, and I like that you showed that species that has a long lower stage actually don't have to have this huge conductivity. You read to the currents, but other studies have also shown some other things. You know what other things that can promote this short distance conductivity?
Well, yeah, in East Antarctica at least, they show that the species was self-reproducing. I don't know about the species, but I know that in certain echinoids, the larvae can, there is kind of a larval behavior, so that so yeah, they can adapt their buoyancy in the water column and direct their swimming depending on currents or oceanographic conditions. But it's really difficult it's more difficult to sample larvae, even though it could be possible, and it will really enhance, I think, the reliability of our interpretation if we would have used larvae. But yeah, definitely there is the oceanography and also the biological trace like larval behavior, which could explain the fine-scale genetics.
Have they seen anything about the larvae that they can actually swim down?
Yeah.
Because that could make them to stay in the same.
Yeah, exactly.
Because the currents are not so strong.
No, at the bottom. Yeah. Apparently, yeah. They also mentioned that in the study in East Antarctica, and they showed that they see that actually the larvae were staying close to the bottom of the ocean and not really in the water column, which for sure then they will travel less and if they stay there.
Yeah. Thanks.
Yes. Thank you, Roman, for the presentation. I really liked it, and to look at connectivity and like understanding 3D. Very valuable. Just a question, we talk about large distances, intermediate distances. What is for you a large distance in this study? Because I think it's important that you really define.
Yeah. That's a good question. For me, the large distance here was like the distance between the two main regions we observed, which was about like 500 to 600 kilometers. And our intermediate spatial scale was about like 15 to 200 kilometers, and our fine spatial scale was a few hundreds of meters to max like 5 kilometers.
Yeah, because it's really dependent on your study species. You have a species which larval phase can float for 4 months. For me, a large spatial scale would then, I think they can reach this in 2 weeks maybe, your large distance. So how could you improve your study that you really can look at these larger distances?
Well, the best would have been like to sample all around Antarctica, and then we could really say there is a large connectivity across the entire continent. We focused on the Western Antarctic Peninsula, but yeah, definitely it would be way better to say there is large connectivity around the whole continent, but because yeah, in this case for 4 months, 600 kilometers is not that long. But it was also to complement the study in East Antarctica, which they used a large spatial scale of like 1,000 kilometers and a small one of like also a few kilometers. So we decided to introduce a bit of an intermediate spatial scale, even though it's dependent, of course, on the scale we are using. But yeah, to see a bit more what's happening at like an intermediate spatial scale.
So I'm a little bit confused because you don't find any population structure with a large spatial scale based on your PCOA, based on your ANOVA, based on your FSTP values. And then you show your FSTP values on a smaller scale, but still 0.001 your FSTP. What does this mean, this 0.01?
Yeah,
it's significant, but
I think it's difficult. We discussed that a lot with my supervisors because I was like, it's so small, it's significant, isn't it like a statistical bias or something? But they were like, that I think it's just like, it's very small, but we have quite a high number of markers, so probably the statistical tests are able to detect a very low differentiation, but still significant. So I think it still has a biological meaning that it's significant, even though it's very low, it still means something, and yeah.
Yeah, there's still a lot of gene flow happening between this finer scale and the finer scale, or not, based on your efforts?
No, definitely there is gene flow happening, but probably less, and we can see that the current may limit, but it's not avoiding gene flow between those locations.
About these currents, so you look at the currents, the pattern, and then you look at the genetic patterns. Is there a test where we can we test the effect of ocean currents on genetic differentiation? How would we do that? Is it like a method you can think of to really like link both?
A method to link really both, like apart from oceanographic modeling, or because we also discussed about that, and I think that it would be really interesting to use like kind of biophysical model, where, for example, we build a model when we put a particle in the water, given the 4 months pelagic larval stage and the currents and the oceanography, we put some parameters and we see, okay, where is this particle going and stuff, and that's why I think it's next study should probably combine those to really better understand the influence of the currents and stuff in connectivity patterns.
That's a good method. So you start with your introduction that you're mainly looking at connectivity, you want to understand connectivity, and you use this species as you selected this species to understand connectivity. So how would you then generalize these findings for other genera or species in Antarctica?
Well, it's a benthic species, so we could with a long pelagic larva stage, and we also selected it because there is more and more studies showing that benthic species with a long pelagic larva stage were forming open marine populations, but more and more studies are showing that it's not always the case, even especially at spatial scales we didn't expect. So we also used this species because it was like to test this assumption of, okay, we have benthic species with a long pelagic larva stage, we expect high connectivity, but how is it really going also at other spatial scales? So it's like to test this assumption of large scale of connectivity in this benthic species. So yeah, we can generalize this finding probably for benthic species with pelagic larva stage, but of course not for pelagic or other species.
Thank you for good answers.
Thank you.
I'm sorry we don't have time for any questions from the audience. Nico. Okay.
Sorry for being.
No worries.
My apologies. But I've seen 90% of the presentation was really great. I really enjoyed how you presented and addressed the value and everything. And this topic is amazing, actually. I really love it, and I could make lots of different questions. But if I have to select a couple of them, I would tell you that if you have to choose in between short scale dispersal for this species or long, larger spatial scale dispersal, according to the biology of the larvae and your results, which one did you choose? Because 4 months in the water column is a lot.
Yeah.
Are you sure the larvae stays there 4 months?
Well, yeah, 4 months is like the longest that it can stay. It's to the literature.
Yeah, but at what temperature?
At what temperature?
Yeah, what temperature were those larvae maintained in the lab? Or?
No, sorry, we did not have larvae samples. We were only working with adults, so the DNA was extracted from adult specimens. So we.
Yeah, but you're thinking that the larvae stay 4 months in the water column.
Yeah.
Why do you think
that? I mean, because it needs time to develop and settle until it becomes an adult.
Usually larval development takes less than that.
Yeah, but because we are in Antarctica, so it's very cold and the metabolic rates are very low, so it's it's longer than in the temperate and tropical species of echinoids. Yeah, definitely, because the cold environment plays.
Could make sense, but along that time there could be a lot of mortality.
in the
Yeah.
moderate increases with
With the-
planktonic larval migration.
Yeah.
And also the larvae needs to feed or not. Is this larvae planktotrophic? Do they feed on phytoplankton or do it have its own reserves? Do you know that? Because that's two people.
Yeah, licitotrophic versus planktotrophic
Exactly.
larvae. Yes. I don't want to say anything wrong. I would say that they are planktotrophic larvae, so they feed on plankton in the water column, but.
Yeah, 4 months feeding on phytoplankton in Antarctica, that's a lot.
So they are probably licitotrophic.
Yeah, I would say that point. I think it could be important.
Yeah.
Because it could be 4 months, but staying at one single place that is particularly productive. But for that, they should have larval behavior, as you pointed out. Also vertical migration.
Yeah, vertical migration and stuff.
But you are not sure of that, right?
No. I mean, it has never been proven for this species, but it has been observed in other echinoids, so probably we can infer that this is also happening, the larval behavior and vertical migration in this species in Antarctica. Yeah.
Okay, thank you very much.
Thank you.
Okay, thank you very much.
Thank you.
So our next presentation is by Thesis. Oh, look. No, hold on. Clicking on the wrong one. Sorry. Oh, it's actually the wrong year. Sorry. Sorry. Hold on. Sorry, Lina, sorry. Okay. Okay, you can start, 15 minutes.
Hello everyone, my name is Lena. Welcome to the presentation of my thesis, investigating the Sigma-Fin Devil Ray aggregations at Ambrosio Seamount in the beautiful Azores. I did this project under supervision of George Bunch and Bruno Massena. Let's dive into it together. Mobula tarapacana is a filter-feeding elasmobranch with a circumglobal distribution. The species is hard to observe because of their long migrations and oceanic habitat. It is classified as endangered by the IUCN Red List. Worldwide, there are only a few known aggregation sites, and one of them is Ambrosio Seamount in the Azores. Here, we have aggregations of dozens of individuals during the summer months. Now, disruptions of these aggregations can have population-wide impacts, which makes these aggregations important hotspots for conservation efforts. The environmental drivers of these aggregations are still largely unexplored in the Azores, and we have no information about the size or the sex structure of these aggregations at Ambrosio. This can give us important information about the life history of the species and also the ecological role the site plays for the species. And while the predictability of these aggregations is a chance for us for investigation, it also makes these aggregations more vulnerable to human disruption, for example by ecotourism. Ecotourism started as a small industry in the Azores in the 1990s, but then rose significantly after the introduction of shark and bait tourism in 2011. A study in the Lingala Reef that was working with Mobula Alfredi has shown that repeated disturbance by humans can actually lead to long-term displacement and also changes in behavior. So an increase in tourism raises some concerns about its sustainability. So we wanted to look further into that, which is why we were looking at Baixo de Ambrosio Seamount in the Azores. It is located 3 nautical miles off the easternmost island of the archipelago, which is Santa Maria. The seamount rises to a depth of 48 meters. And just this year, an order has been released to restrict the number of vessels that can go there to 2 vessels that can carry a maximum of 14 people, and they are not allowed to anchor, they have to attach themselves to one of 2 mooring lines. So now to try to build a baseline about the structure of this aggregation and to also investigate the effect of tourism, we raised several aims for our study. The first one was to determine the size structure, the maturity status and the sex ratio of these individuals making up the aggregation at Ambrosio Seamount. I wanted to identify the environmental drivers of Mobula tarapacana abundance and also investigate the effect of human presence on the abundance of these rays. Now, how did we do that? For the size structure, we used two non-invasive systems, the diver-operated system, short DOF, and the laser photogrammetry system. For the DOF system, I used two cameras that were mounted on a bar 90 centimeters apart, and they were facing towards the center, and like that we created an overlapping field of view so we could see 3D and measure 3D. I had to calibrate the system with a calibration cube, which you can see here, and I analyzed the videos in Thesis, and I basically determined the relative position and orientation of the camera. And then I created camera files that I could use then in Event Measure to measure the disc length and the disc width, so from tip to tip, of each individual. I conducted 16 dives between August and September 2024. Now for the laser system, I used a camera that was mounted on a steel plate between two lasers 25 centimeters apart. We chose green lasers because the light has a further reach in water than, for example, red lasers. To measure the rays, I was positioned directly perpendicular, either above or below the ray individual. I accounted for lens distortion in Adobe Lightroom and then measured the individuals in ImageJ by using the interlaser distance, a scale for the measurement. The laser system was calibrated once a week, and each individual for both systems was measured 3 times. So after data filtering, I was left with 48 individual measurements, and because of non-normality of the data, I used Wilcoxon tests to find differences in the size between males and females. And the only significant difference I could find was for the disc length, with females having a significantly larger disc length with 2 meters than males with 1 meter 85. And we could record disc widths of around 3 meters 70. Now, for the maturity status, I used a published size at maturity data, and I classified all the individuals below this threshold as immature and above this threshold as mature. And I excluded all the individuals that fell within a 50 to 95% confidence interval to not bias my results. I was left with 45 individuals, but I couldn't find a significant difference between the number of mature and immature individuals, and also no effect of the sex on the maturity status of an individual. Now looking at the sex ratio, for these individuals, I also couldn't find a difference between the number of males and females, but I also did some on-site observations during 28 dives, where I determined the sex ratio of the biggest mobile group that was present, which left me with the identification of 142 individuals, and that resulted in a significant female bias sex ratio of 1.9 to 1. Now what does that indicate? So we found some indications that we have female biased size dimorphism, which could be attributed to the higher energetic needs of the females that they need to, for example, support their offspring. We couldn't find significant difference in disc width. That might be because of the positioning of the wings, so maybe they were not always fully extended, as you can see here, even though we tried to find frames where they were fully extended. We found indications for the presence of mature and immature individuals. So Ambrosio could be an important developmental area for immature individuals, but also maybe an important socializing area for mature individuals. It's important to mention that my supervisor did visual cluster observations of males over the last 10 years, and he couldn't find any evidence of apparently immature males. So this might indicate that the size at first maturity actually might vary between different regions. We also found the presence of very large individuals, because for now the largest known disc width is also around 3 meters 70. So this also indicates that Ambrosio provides the necessary resources for these individuals to cover the energetic expenses. And even though we found like a small segregation in time, so like more small individuals in the beginning, we couldn't really find a real size segregation. And it's also important to point out that these small individuals were measured with our laser system, and I conducted an error analysis, which I'm happy to discuss later, which showed that the laser had some higher error than the DOF system, and was very sensitive to laser misalignment. Now for the sex ratio, we found indication for a female biased sex ratio, which might indicate sex-specific habitat preferences. And a female bias has also been observed for Mubula tarapacana aggregations in Brazil at the São Pedro and São Paulo archipelago. Now, why is that? Maybe due to different energetic needs. So females, for example, during gestation, have high energetic needs, so they might be very dependent on local productivity. And for example, during the study period, I could observe some females that might have been pregnant. So maybe Ambrosio is an important gestation area. Another reason could be differences in reproductive strategies. So some studies that were conducted in the Maldives, also on Mubula alfredi, have shown that females show a high site fidelity during mating to very productive areas, while the males are more mobile. So maybe Ambrosio is also an important mating ground. A last reason, of course, could be a bias in observations. Maybe female individuals are more curious, come closer to the divers, which would have made it easier for me to see them and to count them. And even though I tried not to double count individuals, we didn't really do individual photo identification, so I might have double counted individuals, which could have inflated the sex ratio. But overall, if we have a higher presence of females, ecologically that might indicate that females at this aggregation sites are much more vulnerable to disruption. Now that we have looked at the structure of our aggregation, we also wanted to know what are the drivers. And for that, we used a different non-invasive system, the Observed Modular System, short OMS. This system consisted of a camera, a light sensor, a battery, and a controller plate. We enclosed that in waterproof housing, and the system recorded a 5-minute time-lapse video every 15 minutes. So a picture was taken every 5 seconds for 5 minutes. I deployed the system at a depth of 30 meters at one of the mooring lines, and this gave us very high autonomy. We were not dependent on boat schedules, on good weather conditions, and we also got regular sampling throughout the day. The system was shut off at night because we couldn't see anything and to preserve the battery life. Now, our data collection resulted in almost 12,000 collected 5-minute videos. I could sample 36 days in 2022, 34 days in 2024. Sadly, in 2023, there was no mooring line installed, so we couldn't install our system. But as you can see, we had some technical issues, which resulted in some data loss. What I did with these videos was to extract the N-max, which is the maximum number of individuals that were seen within a single frame in a video. And over the time of our study period, this N-max slightly decreased. So to test the effect of environmental drivers, I obtained oceanographic data from Copernicus, and also tidal data, moon illumination data, and the Julian day from our studio. I then constructed several generalized linear mixed effect models. I assessed for overdispersion using the variance to mean ratio, and I accounted for overdispersion by using negative binomial models. I also assessed collinearity using the variance inflation factors. I then built a full model and a null model only including the random effect, and I did a manual forward and backward selection process, which resulted in the same final model, which is this one. Now, this final model could explain 59% of our variance, with fixed effects explaining 23%. Fish abundance seems to be the strongest predictor in our model, and as you can see, large fish groups were associated with the higher abundance of Mubula tarapacana. Our model predicted a 6% increase in Mubula abundance for every additional diver. Looking at the tidal cycle, we expect the highest Mubula abundance during rising tides. For the tidal coefficient, which is the difference between low and high tide in meters, we expect a 100% increase from the smallest to the biggest tidal coefficient observed, and for moon illumination, an 85% increase from a new to a full moon. Let's look at this a bit closer. So, Ambrosio is not really a known cleaning site, so the abundance patterns might actually be driven by opportunistic feeding, and so local productivity might play an important role, which could also be indicated by the presence of other large pelagic predators, like for example whale sharks. The fish community that I could observe during my study mainly consisted of barracuda, of jackfish, and also of bluefish. These species are piscivores and feed on juvenile pelagic fish, like Mubula tarapacana can too, because it's not only zooplanktivores but also piscivores. And so maybe the presence of these fish communities indicates favorable feeding conditions for both groups. On the other hand, joint feeding has been observed between the fish community and Mobula tarapacana at a different seamount in the Azores. So maybe the Mobulas even rely on this fish community to aggregate their prey. Overall, this indicates that Ambrosio is an important biodiversity hotspot. Our model also predicted an increase in abundance during spring tides, which is when we have enhanced mixing and higher nutrient availability, and possibly also local concentration of zooplankton. So maybe there's more Mobulas abundant during these times because feeding is more efficient. During the moon phases, so when we have a full moon, we have higher light illumination, so maybe the zooplankton aggregates deeper in the water column. We have more diving behavior at night, which we couldn't record, but maybe during the day, the Mobulas come closer to the surface to thermoregulate and also to rest. Of course, no model is perfect. In the future, we could maybe consider taking in situ measurements, and also our on-site observation with OMS observations indicated that we had a high number of misdetections. So for example, using a 360-degree camera in the future could reduce this number. We had some problems with our camera system, but we already have a new one that we will test this summer and that will hopefully resolve these issues. The higher predicted abundance with an increase in divers might be curiosity driven because Mobulas have been shown to have high cognitive abilities and also social intelligence, and maybe they are attracted by the bubbles of the divers. It's also possible that we didn't see a decrease because we didn't reach any critical threshold, because the tourism is very small scale and there's a code of conduct, so divers are required to stay on the line and not directly approach the Mobulas. We might still have negative ecological effects, like for example long-term displacement, but we can only test that if we do long-term studies. For the future, I would recommend also combining our study design with acoustic and satellite telemetry to find out more about site fidelity and connectivity with other seamounts. Now to give a quick overview of our results, we found indication for female-biased size and sex ratio, which might indicate that females are more vulnerable at the site. Moon and tidal dynamics seem to be important environmental drivers, and local productivity might drive these aggregations. The associated fish community seems to have a high importance also for the Mobula tarapacana community. We couldn't find any evidence for diver-driven displacement, but the long-term effects still remain unclear and require further monitoring. But overall, Baixo de Ambrosio seems to be an important and critical habitat for Mobula tarapacana, and it's important during conservation approaches that we see the bigger picture and not only this one species. Thank you very much for your attention.
Who would like to start?
I can start this time. Thank you for a very nice presentation. I was wondering a little bit about the size structure of the population. Do they know anything about the growth, the individual growth of males and females? Is it the same?
Well, females mature at a larger size.
Yeah, but the growth, how much do they grow per year?
Per year? I think this is still very, very understudied because also to assess the maturity status, for example, you need that individuals because you have to look at the ovaries, and so I think this is hard to assess. And for example, for this area, for the whole North Atlantic, there's zero information on that.
Yeah, but if they grow, okay. What would you say then that you find more larger females?
Sorry?
If you find more, the females were larger, if they grow at the same rate, what would that indicate from a demographical point of view?
Okay. So overall, females can reach larger body sizes because sexual size dimorphism is known for elasmograms, because the females have higher energetic demands, for example, for supporting the offspring. If you have a high amount, for example, of bigger females and many small males, maybe the female also has a better fitness, because if they have a better fitness, they have a larger body size.
What about age? The age structure?
Well, then I would suggest that the females are older than the males.
If females are older, what would that mean for the sex ratio?
For the sex ratio.
Yeah, you have biased sex ratios. And if the female becomes older than the male, wouldn't you have this result of the biased sex ratios?
Sorry, I'm not sure if I understand the question.
If females survive for a longer time, you will have an excess of females. That would explain your biased sex ratio as well.
Yeah, and of course.
But of course, if you don't have any indication how they grow, it could be either way. Sorry. Yeah.
Thank
Thanks.
you for your question.
Yeah. So thank you for this really attractive presentation on research. So I missed what are these aggregations?
Okay.
What is it and why do they aggregate?
So there is.
Completely new in this topic. So.
Overall, this is a super understudied species. In the Azores, this is the first study ever looking at the ambrosio aggregations. So there's still a lot of uncertainty. We know that they are only there in summer. These are the facts. They come in early June, they leave in late September. They probably go south, probably to Brazil. No one knows exactly why. Which is also why we do these studies, to find out, is this mainly a mating ground, is this mainly a breeding ground? But there's still a lot of uncertainty, which is also why I want to combine this, for example, with satellite data, which would require some time, but it would give more insight if all the individuals, for example, come and go together, if they come back every year, if it's the same individuals, and also where exactly they go, to also relate these different aggregation sites and look further into why exactly they come to these aggregation sites.
So you look at the aggregated individuals, and then is it going to go to generalize these results to the full population? Can we generalize the results from these aggregations to the populations? Why not and why can't we?
I would say you can't, because every aggregation can be very site-specific. So for example, a study in Hawaii showed that depending on the feeding location, you either have a female biased or a male biased sex ratio. Or for example, it depends a lot on the goal of the site. So the main drivers have been shown or are hypothesized to be productivity or cleaning. So for example, during low food availability, these individuals have been shown to more go to the cleaning sites and have a higher abundance. And then when feeding is more efficient, they tend to go to the feeding sites. So I think it's very site specific, but then what is important is that there's high connectivity. Like, these individuals move southward, they don't stay in the Azores. So if we impact this population at one, or this aggregation, sorry, at one point, it will impact things at another site.
So you found this biased sex ratio, give a lot of hypotheses, also the hypothesis based on literature, what is the most likely one? And if not, if there's no information, how can we test and differentiate between these hypotheses that you mentioned?
So I would say the most likely one is that it's a breeding area and maybe a mating area. We could test that. We actually plan to do that this summer, and by really assessing if the females are pregnant by using a new technology. And so this could give clear indications about that. If we don't find any pregnant females, it's for sure not a gestation area. We could also assess mating scars in the future. I would like to incorporate that in future studies. I didn't have enough time. But if you have fresh mating scars, then you have clear indications that it is a mating area. If you have none of the two, you can think further.
Okay, so let me see if I understood this. You have basically two different methodologies. One where you devise this technique to measure the race.
Yeah.
And then you had data from cameras from 22 and 24. Is that the case?
Yes.
Okay. So for the first part, I think it's an interesting methodology you're developing to measure the states.
Mm-hmm.
Okay, you characterize the population, and I wrote here 56% females with your first approach, is it?
Mm-hmm.
And you found that to be a very unbiased sex ratio?
No, I did on-site observations in addition to that.
Okay.
So like from the individuals that I measured with my two systems, I couldn't find a significant difference. But I had to exclude a lot of individuals that I saw during the dives because the measurements were not good, because the conditions were not good enough, which is why I also wanted to include my observations during our dives, which is when I could really assess the sex of every individual, which also gave me a much bigger sample size, and that gave me the sex bias.
Sex bias, all right. So, and there you mentioned you could have oversampled, so you could have counted one individual more than once.
So to be really sure about that, you would really have to do photo identification, and then assess like how many individuals you double count, which could also give a better insight into site fidelity, because if you photograph the same individual every single day, then you don't.
Okay, and I was going to ask you that, but you answered that you didn't do photo identification. Is there a database of, is there like sharks, marks that can be used to recognize individuals?
Yes, they are belly pattern. So the belly pattern is like a fingerprint, so you can just have to take a shot from the bottom, and you can identify every single mobula with 100%.
Because that would be a very interesting thing to solve a lot of your problems with connectivity, you know, where they go, where they're coming from, and so on.
There is actually a project in place that is only focusing on that, so it would be interesting to combine that.
Okay. So, and you mentioned in the beginning that it looked to me that at the beginning you had the purpose of checking if tourism could affect this population. But there's no evidence of that. No? Do you have any evidence that there is harm to the population of having tourism?
Not to the specific one, but there is evidence for other species. As I mentioned, for example, for Mugula alfredi in the Maldives, or also in Australia, there were studies that found long-term displacement. So after the tourism increased, the number of rays that came back to that site decreased dramatically, which also could be dependent on other factors, but these studies clarified that they tested the effect for tourism. And also, for example, it might affect the behavior. So some studies have, for example, shown that if a diver directly approaches a mobula, they change their behavior, which can also like disrupt feeding activities, which might also decrease their overall fitness. And I think the code and conduct that is in place at the moment and the restrictions keep it at a good level, because divers can't approach the mobulas approach the divers, and we have a very limited number. So this is also why our study says, like, for now, we can't find evidence of a negative impact, and we think that the conservation measurements that are in place should be sustained. But yeah, for other species, there has been an impact. Also, for example, for boats, if you have an increase in boat numbers, because the mobulas sometimes come quite close to the surface, so there's also a higher risk of injury.
Okay, just one curiosity question I'll put at the end, but I wanted to ask you for the 22-24 data. That model, the 22-24 data, where you have all of these variables, was year a variable, because I couldn't see it there, or did you put together 22 and 24?
I put it together, also because I had like these data gaps in my data, so I don't have exactly the same times between the two years. And also because of the limited number of data, I didn't want to introduce another variable, but we will sample again this year, and I hope that after the season we will do a new model where I want to include more.
So you have no idea if the numbers between 22 and 24 stay the same, increase, decrease, you don't have that?
No, for now it's overall just...
Because it seems to me that the most interesting thing of what you've done is the repetition of the time of this standard methodology. That will allow you to monitor the population, combined with individual verification and things like that. Yeah. Then the curiosity question is, how many manta ray species are there in the world?
In the world? There is 9. There's 7 7 devil ray species, and then there's Mugula alfredi and Mugula pedostris, who have been classified as manta ray, but now they all belong to the family Mugulidae.
Okay, and in the Atlantic, how many?
In the Atlantic, I'm not sure, but Mugula tarapacana is the most abundant one in the Azores. Sorry.
Yes, to be brief, very nice presentation. I really like how you presented a nice combination of video, speech, and so on. It's a very nice science, and very nice stats. I think if I were you, would focus more in those events that you carry out there. And usually that variable that you have there was not significant, I think, but moonlight with the package center, right?
Moonlight was significant.
Yeah, and moonlight was significant.
It was significant.
And can you tell us something more about it? When there were more man-hours?
Well, there were more mobilers expected, according to model predictions, when there was a full moon. Yeah, from a new to a full moon. And my idea is that this is obviously also related to upwelling events, to mixing of water masses, food availability, and also this is a very big hypothesis. I don't want to, it's just an idea I came up with. Because of the diurnal migration of the zooplankton, when we have high moon illumination, another study has shown that 55% of all pelagic species dive deeper, and mobulas need to thermoregulate and also to rest. So my prediction is that maybe in the following day, they also stay around more in the shallower areas around the seamount, which is when we can capture them with the camera. That might be contributing to the higher variance, but it's a big hypothesis.
That's a long shot, man.
Yeah.
Yeah, okay, thank you very much.
Thank you.
Thank you. Okay. Our last presentation is Naeem.
Hello everyone.
Okay, everybody ready? Okay, go ahead.
I am presenting my work on phytoremediation potential of chlorothalonil cell for the uptake and accumulation of two pharmaceuticals, carbamazepine and gemcitabine under hydroponic conditions. I did my work in extreme bio group in Sisima, University of Punjab Farm. This is a brief outline, and here is the background of my study. As we know that pharmaceuticals are used for the prevention and treatment of diseases for humans and animals, and because of their benefits, their global consumption is on the rise, and everyone is using pharmaceuticals. So they are entering in the environment from multiple sources, directly releasing from the industries, as well as excretions of humans and animals. So there are multiple sources and routes towards the environment. So when they are entering the environment, they are having some negative impacts on the environment. So when we want to treat them, there are two challenges we face. They are persistent, and even they can bioaccumulate in the organisms. So due to their persistence, normal conventional treatment methods are failed to treat pharmaceuticals usually, and when we employ advanced treatment methods, these treatment methods are usually expensive, and sometimes the treatment methods also produce secondary pollutants. If you are using chemicals, then you are also producing the secondary pollutants, which is another problem. So at this time, it's important to find a useful method. So I have selected here two important pharmaceuticals, carbamazepine and gemcitabine. So why I have selected, there are three main reasons. First one is they are frequently prescribed pharmaceuticals, and the second, they were detected at high levels in postural diurnal estuary. So it was an important reason to select them. Third one is that they have toxic effects on the organisms, like they can cause reproductive disabilities, hormonal imbalance, and neurotoxicity. It is already established. So when coming to the possible solutions, here we are focusing on the nature-based solutions. So phytoremediation is the one in which we employ plants to degrade or treat the contaminants. So there are few advantages of phytoremediation. It is an eco-friendly technique, and plants, as we are using plants, they are easily available and we can select from many options. And if we are growing plants to treat something, they are also adding visual appearance, like visually they are appealing. So when we talk about the phytoremediation, it's very important to select an ideal species. So this study is basically to assess that if Plantago is a good species for the phytoremediation of pharmaceuticals. So when we are selecting a species, it is important to know that if they have the high uptake ability, or it can tolerate climatic conditions, different climatic conditions, and how much biomass it produces. If any species producing high biomass, it is good for the phytoremediation. And also we need to see the root system. If there is a good root system, we can expect that it will extract more contaminants from the soil or the water. So also it's very important, if any plant is extracting any contaminant, it should have the ability to endure that pollution. So this is already established in the literature that what an ideal species look like. So here I have two objectives. First one is that I want to assess the capacity of P. carnopus for the removal of CBZ and GEM from contaminated water. And after that, the long-term aim is that I want to assess the potential if P. carnopus can be employed into constructive wetlands or not. So the second objective is that I want to study the plant's efficiency in terms of uptake, and then after uptaking, in terms of their translocation within the tissues as well as bioaccumulation. So how CBZ and GEM are bioaccumulating in the tissues is my second objective. So I am hypothesizing that P. carnopus is capable of effectively absorbing carbamazepine and GEM fibrozil from the medium, and it will be a promising candidate for the phytoremediation. And my second hypothesis is that the phytoremediation efficiency of P. carnopus will increase with the exposure time. As long as it will stay in the medium, its ability to phytoremediate will increase, but it is depending on my experimental duration. I have experimental duration of a certain time period which I will mention. So this is my experimental design. I have 4 time periods: 1 day, 3 days, 5 days, 7 days. So the second hypothesis is basically for the 7 days. Like I am hypothesizing that the uptake ability will increase towards the time exposure. So then I have 3 treatments. First one is the solution, in which I have provided carbamazepine and GEM fibrozil in a mixtured form, and then I have grown plants in it. And the second one is the control, in which there is ultra pure water in which plants were grown. And the third one is the abiotic control. It is without plants, but the solution is the mixture of CBZ and GEM. And it is to study the environmental factors and the possible abiotic factors which can also play a role in the degradation of these contaminants. And I want to mention that here I have also added ratpin. It is a fertilizer, you may call, like it will help plants to grow in hydroponic conditions because there will be no soil, so the plants will use this ratpin to grow. This is my experimental methodology. I cultivated plants in the medium, and then they were transferred inside the hydroponic conditions for the experimental duration. And after completion of the experimental duration, 1 day, 3 days, 5 days, and 7 days, I taken them out, and then I took just 1 gram of fresh material and grounded them in the pestle mortar using HCl and acetonitrile. Then I sonicated this material, and after sonication I used centrifugation at 6500 rpm, and then I collected supernatant, and this supernatant was then adjusted in terms of pH. The pH selected was 4 because in the literature it was established that the 4 pH is the best pH to extract these 2 contaminants using SPE cartridges. So after pH adjustment, it was the time to pass this extract from the SPE cartridges. SPE cartridges have they are the small cartridges. They have a solid powder which can extract these contaminants inside it, and then we wash it out and dilute. We took a dilute. After taking the dilute, I evaporated the dilute up until the dryness, and then reconstituted it with the acetonitrile and prepared the vials for the LCMS analysis. After LCMS analysis, I used Excalibur software, and in this software I detected, first of all I detected the peaks, and then I quantified those peaks. First of all, I prepared a calibration curve. This calibration curve was made using known concentration of 268 up to 16 microgram per liter of the contaminants. And then I did statistical analysis and also draw some graphical representation. After that, I calculated percentage renewable efficiency, translocation and bioconcentration factors. They are important factors to understand the phytoremediation. And then I performed crystal values test to study the significance, and this was followed by Dunn's post hoc test, and to study the significance between the days. So these are the few formulae which I employed during this study, and then I got these results. In the initial, at day 0, I used 10 microgram per liter of these contaminants. In the mixture, 10 microgram of carbamazepine plus 10 microgram per liter of gemfibrozil. At day 0, we have we have this concentration, but after day 1, like you can see the decrease in the medium. You can see the medium has the color of red, and the abiotic control has the color of yellow. So they are both decreased, but medium was like in which plants were grown, it was decreased most. And when we see from day 0 to day 7, we have a significant decrease, 6 times decrease from the day 0 up until day 7. It was like from 10 to 1.63 microgram per liter. And we also have seen some reduction in the abiotic control, but it was not as much as was in the medium in which plants were grown. So the reduction was from 10 to 5.93. So when we see the concentration in the leaves on the day, in the leaves are green and the roots are gray. So when we see in the roots, the concentrations are high on the day first, and in the leaves it is less, and progressively it increased from 0.71 to 11.13 microgram per kg in the leaves, and it decreased in roots from 7.66 to 5.67 microgram per kg. So from here we can conclude that there is some plants driver removal for the carbamazepine. Now we can see the possible reasons behind these results. If we see that in the previous literatures, they described that such kind of species can uptake large amount during initial 4 to 7 days. So same kind of behavior was seen here, like from the medium the plants has uptaken most of the like contaminants during the early 4 to 7 days, and then we have observed that the contaminants are going from like roots towards leaves. So this translocation is basically connected with the neutral charge of carbamazepine, and they have a moderate lipophilicity, which may like enhance the xylem's activity. So in the xylem form, they can go towards the aerial parts. And sometimes plants also use this mechanism of translocation to act in terms of defense, like they use plants transfer the contaminants in the leaves, and then they have the time to degrade these contaminants, as it was discussed by Thesis and al. When we see the gemfibrozil, the concentration was also in the medium was also dropped for the gemfibrozil, and it went from 10 to 3.55 microgram per liter. It is a significant value. But when we see in terms of comparison with the abiotic control, you can see that abiotic control is showing high reduction as compared to the planted media. So in this way we can say that abiotic processes likely have more influence towards gemfibrozil, and also this story doesn't end here. Like we can see some accumulation has been in the plants tissue, like the plants in the leaves we can see, and in the roots there is accumulation. Okay. And these results are going towards like gem proposal can typically go inside the roots, like they have low KOW value, so literature suggests that it will stay longer in the roots, but here I didn't see any significant pattern like it is increasing and decreasing in the roots and leaves without any specific pattern. And most of the reduction was also seen in the abiotic control. So this behavior is very unusual, and these results are not promising for the gel fibrosing. So these are some of the values which also confirm that they are basically comparison of removal efficiency for the abiotic and the biotic factors, and we can see gel fibrosing has almost similar values for both removal, and but for the carbamazepine we have promising results because 84% of removal efficiency was achieved in the planted media. So translocation and bioconcentration factors are also important. As we have seen, translocation was done from the roots towards the leaf. So I already discussed that it is a defensive mechanism, and there is no pattern for the gel fibrosing. It was increased in the start and then it started decreasing and then increased. But we have a very clear pattern in terms of carbamazepine translocation factor is increasing towards the end of the experiment. And the similar situation is for the BCF. Bioconcentration factor is also behaving in the similar way. So the limitations are that I didn't take into account the transformation products. So transformation products can be analyzed, should be analyzed, because if any plant is degrading something or metaboliting it, we have to see if the transformation products are less toxic or even more toxic than the previous version. So it's my suggestion we should go for the environmental factors also and for the transformation products. And now I would like to say that what was the purpose of my study was achieved. Like I have seen that P. carnivus effectively removed CBZ and gem, and these two contaminants were also translocated towards the aerial parts. Both of my hypotheses were met, and in the future we should have a longer time period, as well as like we should need to consider a lot of environmental factors and the real-time scenario of the contaminants. Thank you.
You went a bit over time, but so who would like to start?
Okay, yeah. Thank you. Thank you for the presentation. Thank you for your research. So, to start, what are these pharmaceuticals used for? What do you explain this? So why do people use these pharmaceuticals?
They are used to treat or prevent the diseases.
Okay.
Yeah. So, basically, it is a very advancement in the health sector that they are produced, and now we every time using for these compounds to prevent or treat any disease, even in the animals and in the humans as well. So this is the basic use of it.
And why did you see like those two? You already found to be accumulated in the
Yeah, I described that there were three reasons to select these tools. So first one is that they are frequently prescribed pharmaceuticals, and the second one is that they were detected at high levels in Portugal's by the laboratory, and third one, there are some established toxic effects associated with them. So they can cause reproductive disabilities and hormonal imbalance, etc.
And so in your methods, you always use a combination of those both products, both pharmaceuticals.
Yeah.
Why did you go for that combination? Why not just study
Yeah. So basically, in the real situation, they are coming in the mixture form, so they are not separate. So if we see the real-time situation, they are coming. Everything is coming in the mixture form. Maybe they are two, and in the real, we may have 100, 200 pharmaceuticals coming together. So it was a better idea to go for the mixture instead of making separate treatments.
Okay.
But it has it may have some effect like synergetic, or maybe have negative impacts on the signal. So if carbamazepine is present, maybe it is depressing gene fibrosing, which was not detected properly, and maybe the opposite trend can go, like signal can enhance.
Then one more question. So the idea is that to construct wetlands with this plant.
Yeah.
And then what happens then? So you construct these wetlands and these pharmaceuticals are within these plants?
This is a very good question. Like the biomass, when it is biomagnifying, I have used BCF. Like it is showing that is it the possibility that the compounds with biomagnify, if they are biomagnifying in the plants, then it's a big problem. So we need to treat them further. Like we have to do pyrolysis, we have to do incineration. We need to properly destroy them. So this is a big problem. So, but the problem is that if we are like extracting pharmaceuticals from the water, then we can use this biomass for the energy production, for any other task. So this is another potential study, what kind of potential uses from the biomass can be taken from that. So that's why I'm thinking to it requires a lot of more studies before going directly into the constructive wetlands, but I was just studying the potential if it is possible to do that or not.
Well, maybe I should pick up this last question, which is my biggest worry. We have a very good, well-designed experiment, proper controls, and clearly this plant works. I have a question. Does it float, or you have to have it supported by the sides of your suppose whatever those cups you used? Doesn't float. So my question is, to be effective, it has to be planted, no?
Yeah.
In sediment.
In sediments. Here I had hydroponic conditions, as I mentioned, so there was no soil, they were floating and they were absorbing.
Because it was supported by the sides of
the container.
Okay, of the
Yeah.
container.
Yeah.
But my question is, so you have a combination of the plant and the sediment. I assume it's very fine sediment that you have in these areas. What kind of plant? What kind of sediment?
Yeah, when I was employing these plants, I tried to clean as much as possible with the ultra pure water before growing them in the hydroponic condition.
The biotic control was with sediment.
No. It was like the system was totally same but without plants. Yeah, abiotic was
what
without.
What did you put inside those?
The similar solution. I was studying the possible photo degradation, possible environmental factors playing to degrade
the same solution. So you just let it stay.
Yeah, for the similar period to see if there is a plant mediated removal or if there is any other factor, light or any environmental factors, dissolved oxygen is playing a part.
So you have the problem of the sediment. My problem is, of course, you cannot clean the water of the river.
Yeah.
But this goes to the river through sewage, no?
Yeah.
So doesn't it make more sense to apply this in controlled facilities, not wetlands, but in controlled rooms associated with the sewage treatment plants, which could extract not only these two components, but the thousands of chemicals we put in the water, do it in a more general way. Does it make sense to propose controlled wetlands where you're going to grow highly contaminated plants?
Yeah, it is very important. Like here I was just assessing that if this plant species able to accumulate or uptake these contaminants or not. If it is not uptaking, we cannot employ that in the broader area. So this study is only focusing on the potential of this species. And here we have seen some potential, then we can go for the further studies in which we can involve soil, possible fertilizers, possible contaminants, changing in the environmental factors, all these. So under hydroponic conditions, they are growing at 25 degrees Celsius, 12 hours of night and 12 hours of day. But if we are changing certain conditions, definitely they will perform differently. So this will be a
From hydroponics, if it's a plant that doesn't float, it will work in the real world.
Yeah.
So anyway, very nice work, but I hope that engineers pick up this and develop the structures where this can be applied, because for now this is very interesting, but I see it very hard to be applied in a real life situation. So that was my questions.
I have a short question. Well, is there other studies where they combine different species of plants to have this system even more effective? Because maybe one species can take away some of them, and another species will be good at taking away others. Is there any such studies that you see?
Basically, for the carbamazepine, we have some studies, but for the gemfibrozil, it was a totally novel. No one has, up to my knowledge, no one has used phytoregulation to treat gemfibrozil. And in the nature, it is the similar condition as you are telling that a lot of plants are growing as it was found in estuary. So we are seeing that around the estuaries, these kind of plants are present, and if they have the potential to uptake and treat automatically without one further treatment methods. So if they are automatically doing this, so then it is very important. And the second point is if they are uptaking, accumulating inside this, and some people, I have seen some literature that it is used in salads. So if the contaminants are inside, this study has another perspective, like the contaminants are inside and you are using as a salad, then it's a problem and it can enter the food chain. So yeah.
So the part picked up by the river.
Yes, thank you.
Yeah, very nice presentation. Very good and friendly, and yeah, it was very well explained. Although I missed that you saw a little bit in that slide presenting the dependent variables. Yeah. You were going to just pass it like.
Yeah.
The equation of each one. I don't know which variables.
On the formula.
Yeah, with the formula.
Okay.
We just went through that without not stopping at all. So I missed that.
So
yeah, this is that. Yes. Okay, now I see.
Yes.
And no, but I have one question. This is not my topic, so I don't know, but it's just curiosity. The transformation products, no? Is it common that you get a transformation product, a product produced by the plant when it uptakes the first contaminant, that this product, this secondary product is even more toxic than the first one? Does it occur often?
For the pharmaceuticals, I'm not sure, but for the biocides, it happens. Like I have seen the literature that biocides, if they are treated, they are transformed into more toxic products. So this is another problem which I have to counter. Yeah, but these two contaminants, they are very persistent, and I'm expecting that they will not be degraded easily by the plants also. So they may be present in the original form, and we have to counter with biomass. So biomass need to be pyrolysis or incinerated, or you have to use in any kind of energy production in the anaerobic processes. So this is the thing.
Okay, thank you very much. Any questions? Thank you for a very nice presentation. I just have one more question. I remember you mentioning, one of your hypotheses was that that the uptake potential would increase with exposure time.
Yeah.
I was just wondering why.
Basically, in the first day, plants may be acclimatizing with the toxicity of that contaminant. So after passing of a certain time period, they will be easy and they can accumulate more. Yeah. So that's why I hypothesized, and it is based on the literature, for the short periods. I'm not talking about for the longer periods, and it is valid only for my period, 1 to 7 days. So I am expecting that in the day first, the removal efficiency will not be as much as on the day 7. Yeah. So this is basically based on the acclimatization period.
Yeah. Okay, thank you.
I have a question. the concentration of these pharmaceuticals that you use in your experimental setup. How did you choose that? Is it typical of what is found in the estuary, or did you make it up? Or
it is I have seen the literature, and the 10 microgram per liter is usually the concentration which is found for the pharmaceuticals. So I selected the same. So because previously I was selecting like 1 milligram per liter, then I realized that it is not possible that 1 milligram per liter is coming in the real situation. So this concentration should be lower. So I tried to find something, and then with my supervisor we decided that as we have seen in the previous literature that mostly these kind of contaminants are present in nanograms up to micrograms, then it is suitable to take 10 microgram per liter. And we have to take into consideration the limitations of our machine. Like if I'm using very low concentration, maybe it is not detected by LCMS. Maybe it will not be extracted properly. So that's why it was the feasible one, and I studied it before employing this value on a smaller scale, like using 2 to 3 plants, and then I see that it is easy to quantify this, so then I started this way.
Well, thank you very much.